对于脑的认知有很长的历史。在过去100年到200年内，是神经科学研究的主要时期。我们北京大学对于神经科学的教学和研究也有相当长的历史。美国的 Patrick、Lori McGovern 夫妇捐款资助北京大学成立脑科学研究所，对于我们北京大学凝聚一批通过不同的交叉学科的研究途径研究神经系统有很大的促进。
10年左右，我跟随着劳毅老师，参与了我们麦格文的早期创建工作。
我记得10年前，当麦格文先生和麦格文夫人到北大来推动这个项目的建设的时候，各个方面呢都感到这是建设北京大学交叉学科的一个很好的机会。
有做细胞生物学的，有做神经生物学的，有做神经环路，还有做神经电生理的，还有我们做临床转化研究。
当时麦格文夫妇和北大在灵湖县签订成立麦格文的这样的这个协议，那时我刚好是在北大面试结束，所以加入麦格文这个其实还很有渊源。
北大最聪明的一帮家伙，特别是说生物学、心理学的老师，因为研究所的存在，大家有很多交流的机会。
当时我记得大概就是10个人左右，经过10年的发展，现在 PI 呢有非常多，领域呢也增加了非常多。
这个脑是最复杂的，所以呢，它的研究可以从心理方面、生物方面，或者呢是通过化学的这个手段来研究。
如何能够研发出来更好的、更全面的可视化的手段？
建立了世界上首个清醒猕猴的双光子成像的实验平台，
有足够的能力来同时记录大量的神经元的活动。
我们通过研究人脑本身的时空信息处理规律、存储规律以及认知的规律，
推断他人的策略，猜测他人的目的。
我们这个研究所很多 PI 说做更加微观的这个神经生物学、神经生理学方面的研究，在宏观的层面也有做理论、做计算模型的老师，
用数学建模或计算机的方法研究大脑信息加工的基本原理，
结合抽象模型和脑磁图研究决策中的偏见，
把我们在基础研究的问题和临床老师去沟通，在病人上进行测试，
在治疗方面进行了一些初步的探索，
来帮助我们患者解决他的一些痛苦，
会引领我们整个的医学学科的快速的发展。
神经科学从整体而言，既是思想驱动，也是技术驱动。它最终的发展还是要关注人，他们怎么样治愈人类的脑疾病，怎么样提高人类的认知功能。
那我们麦格文研究所最大的一个优势，它就吸引了不同背景的科学家们来一起努力工作。
Brain science, we need multiple of the studies.
无论是年长的教授还是年轻的研究人员，大家都能够在一起直面科学问题，
去定义问题以及解决问题的这个思路，大致上来说是非常相通的。所以你永远不知道在下一刻大家之间的交谈最后会引发出来什么。
那我们每个月呢都会组织一些午餐会，在这样的一个过程中，就非常好的促进了大家的思想交流和学科交叉。我们利用李玉龙老师开发的分子探针来研究疼痛当中地质释放的一些问题。
怎么样能够看得深？不光是单单的是这种化学的探针，自己能够就要有这个更好的物理成像的手段。
我们研发的新的脑磁图呢，可以深入到大脑的深部可以看一些动态的神经，可以推广到神经疾病的方面研究。7T 呢可以达到亚海米状态，进入介观层次，可以看大脑的一些分层、皮层的成像。
北京大学培养学生是有它的特点的，
充分尊重学生的个性，
按每个人的特性来选择最适合他们的 project。
我还特别呢会关注
对于女性研究生她们的成长，特别注重对于女生的持续不断的激励。
鹿鸣书院现在一个非常大的特色就是本科生从1年级就和老师有一个非常密切的互动，他们从这个学习中能够了解到什么是神经科学，什么是真正的科学实验。
从一个学生的角色转换成一个老师的角色，很荣幸能回到母校来从事科研工作。
神经科学就是在这种不断的挑战与被挑战中不断的发展，不断的碰撞出思想的火花。
可以把北京大学的这个生命科学、心理学还有这个临床医学能够很好的结合在一起，应该说是一个非常好的制度设计。
整个过程呢，我可以总结为从耕耘到收获吧。
如果你想走得更快的话呢，你一个人就好了；但是如果你想走得更远的话，那么你需要更多的人加入。
神经科学是充满着很多的不确定，但也是充满着很多的希望。
随着国家对于科研的这样重视和投入，以及科技兴国的这样一个战略，所以我觉得未来是大有可期的。
人工智能与神经科学的融合将会促进我们对自身以及对世界的理解。
类脑计算的突破呢，会带到人工智能的突破。
我们能发现更多的靶标，那更多的新药会层出不穷。
用神经科学的方法去解决儿童发展或者是发育障碍，
对于各种神经系统疾病的治疗以及药物的开发。
同一个情况下可以研究一个脑区大多数神经元的活动，
能够在神经认知机制、神经成像技术、神经疾病治疗等多个方向取得突破性的进展。
将近10年来，通过交叉学科的研究，对于脑的认知和脑的疾病都有了推动。我相信在未来的10年，北京大学本高本脑研究所将成为全世界一个非常重要的脑研究机构。
Of the Beijing Brain Conference on Autism Spectrum Disorder Quantitative Diagnosis. So autism spectrum disorder is a developmental neurological disorder characterized by deficits in social interactions, restricted interests, and repetitive behavior. It affects millions of individuals and their families worldwide. So a big issue in the field of autism is quantitative diagnose, how to quantitatively diagnose the patient. And this is the main topic of our forum. So we're very honored to invite as our first speaker, Professor Sarah Jane Webb from the University of Washington and the Seattle Children's Research Institute. So her lab uses many quantitative measurements to study how children encode, store, and retrieve information about visual images, such as faces, and how these processes are related to social ability and social disability. So the title of her talk is EEG and eye tracking risk factors in infants with elevated risk for autism. So please welcome Professor Sarah Webb.
Thank you, Dr. Yu and Dr. Yi, for inviting me, as well as the attendees at the Beijing Brain Conference today. The goal of my research group has a focus on using EEG and eye tracking measures to identify neurological processes related to autistic behaviors. Before starting, I'd like to extend a special thank you to the individuals with autism and neurodevelopmental disorders who have participated in our research with their friends and families. I would also like to thank the parents of infants and toddlers who engage in research during the stressful pre-symptomatic period or while seeking a diagnosis. My research is currently funded by the National Institutes of Health and the Simons Foundation. I do not derive funding from profit-focused industries. I have the pleasure of working with fantastic collaborators historically and currently. I trained with Dr. Nelson and Dr. Dawson, and the work I present today includes highlights from their work as well as that of Dr. Fred Schick at Seattle Children's Research Institute, Dr. Emily Jones, who is a professor at Birkbeck University and a former postdoc in my lab, Dr. Shefali Jesti, who is the director of child neurology at Children's Hospital of LA, the Baby Siblings Research Consortium, and the Autism Biomarkers Consortium for Clinical Trials. As Dr. Yu stated, autism spectrum disorders is a neurodevelopmental disorder with significant impairments in social communication, social interaction, and increased restricted and repetitive behaviors, as well as alterations in sensory responsiveness, including both hyper and hypo responses. 80% of parents with a child with autism spectrum disorders report having developmental concerns prior to the age of 3 years. Many of the autism risk genes impact early brain development, likely converging on neural circuitry formation. In considering these downstream consequences, these downstream genetic consequences, our work is guided by the theory of Dr. Mark Johnson, of which interactive specialization proposes that postnatal neural changes occur as regions interact and compete with each other to acquire the role in new computational abilities. Thus, some cortical regions begin with poorly or broadly defined functionality, such that these undifferentiated neural activation is more likely early in development across a range of stimuli and task contexts. With development, regional activation is tuned by its own activity as well as the patterns of connectivity with other regions. Increased neural tuning or specialization in response to social information and social interactive demands results in a selective narrowing of response patterns with age. For example, unique event-related potential responses to upright human faces emerge from more broadly tuned responses that are initially elicited equally well by closely related stimuli such as inverted or non-human faces. As the fusiform gyrus becomes more specialized or finely tuned for faces, it will respond less to non-preferred stimuli. That tuning is environmentally dependent, as our brain specializes to process the faces that are most present in our community and our environment. This tuning is further influenced by the systems that interact with this information, that is, frontal circuits that influence approach and withdrawal behaviors. This will form circuitry to support the use of face information to guide behaviors, and auditory and speech integration, which is used in multimodal processing of phonemes and word perception. These interactive demands in early development create specialized cortical regions, of which those are disrupted in autism spectrum disorders. Human infants learn better from social interactions, and neural development and behavioral learning occurs within that social world. Infants are reliant on human caregivers and thus exposed during most waking experiences to social, visual, auditory, and tactile experiences. In autistic individuals, genetic alterations change the fundamental underlying neural infrastructure of this brain development. Autistic behavioral differences, by the time of diagnosis, represent multiple years of the complex interplay of genetics, neural development, and experiential environmental factors. Geschwind and Levitt in 2007 referred to autism as a developmental disconnection syndrome, in which the early neural circuits are altered such that expected experiences are encoded into less functional neural circuitry. These disrupted developmental pathways in sensory processing and attention further shape and alter neurodevelopment over time. Dr. Tarwaska from Yale University states that behaviorally, the depth of processing of social stimuli is atypical in infants who later develop autism spectrum disorders. This early alteration, the type of attention, increased looking at non-facial stimuli or decreased looking at the relevant parts of the face, shapes the way in which the social brain acquires its computational abilities, altering the spatial extent and tuning of the brain. Decreased or inefficient computational abilities will, over time, make social information less informative and less relevant for complex behavioral processes. By 18 months of age, the red flags for autism diagnosis reflect altered sensory and attentional systems and the resulting behaviors. These early signs, such as decreased eye contact, lack of attention to others, result in difficulties with joint attention and social referencing. We think of social attention, social motivation, and face processing or face perception as the building blocks of complex social processes that result in joint attention and thus support shared social learning. decreased or poorly integrated joint attention will reduce this learning. And as seen as these images of infants and toddlers, you can see how much attention to the faces of others, dyadic attention, and attention to objects, triadic attention, influences daily activities of young children. How do we go about studying the early pre-symptomatic processes in autism spectrum disorders? ASD prevalence is less than 2% in the general population in the United States of America, making surveillance studies that include in-person measures difficult to study early processes to get enough children with those autism outcomes. Thus, we turn to elevated likelihood risk groups in which the rates or prevalence of autism diagnosis are higher than the general population. In the medical model, premature infants born less than 34 weeks of gestation age or at low birth weight, less than the 20th percentile of weight, are at elevated risk for autism spectrum disorders and poor developmental outcomes. About 7% of premature infants are found to have a later diagnosis of ASD in a meta-analysis by Agrawal, shown in this graph. The greater the prematurity and the smaller the birth weight, as shown in the graph on the right, the higher the cases of ASD per 1,000 live births, as identified by Talmi et al. In a recent study of infants in Israel. Thus, we can think of medical risk, early birth, and small birth weight as potential risk models to help us understand pre-symptomatic processes. Similarly, in the familial risk model, infants are ascertained based on the presence of an older sibling with autism spectrum disorders. Data from the Baby Siblings Research Consortium, a 15-year consortium with combined data from over 30 research groups working with this population, suggests a reoccurrence rate of 19% in a younger sibling of a child with autism. Dr. Nicole McDonald from the Justy Lab, in a more detailed analysis, has identified a reoccurrence rate of approximately 16% when there is one older autistic sibling, but up to 36% when there are two or more autistic older siblings in the family. Thus, we can look at family studies to help us understand and identify infants that are at elevated risk. Similarly, we can also use a genetic risk model that infants with genetic syndrome conditions related to autism, like tuberous sclerosis complex or Fragile X syndrome, or novel rare variants identified in autistic children, can be followed prospectively. I highlight in this presentation the tuberous sclerosis complex work by Dr. Justy and Dr. Nilsson. TSC is an autosomal dominant disorder caused by a loss of function mutation in the TSC1 and TSC2 genes. It causes benign hematomas throughout the brain and body and has a high penetrance for ASD, with over 50% of children having a diagnosis, as well as a high penetrance for cognitive impairment. TSC provides a unique opportunity to study development, as early diagnosis can occur in utero. So trying to think about how we can use these models to better understand the onset of autistic symptoms, we know that currently within diagnostic centers of excellence, diagnosis of autism spectrum disorders can be made at approximately 2 to 3 years of age. And many of the evidence-based interventions for children with ASD have been developed for this age group and older children. Between 1 and 2 years of age, early behavioral signs are related to later diagnosis, although the sensitivity and specificity of those measures and outcomes vary greatly based on population and the type of measure. Our goal is then to identify measures before 1 year of age that will inform us of early neural circuitry function and are related to these social building blocks. EEG and eye tracking are non-invasive methodologies that are stable across age, meaning we can use the same methodology from birth through the lifespan, and they are temporally sensitive. They are also feasible and scalable for infants. EEG activity reflects synchronized activity of large networks of neurons during resting, induced, or evoked experience, while eye tracking tells us where the child is looking as an index of overt visual attention. And as you see as these images, we can adapt these methods to be used with these very, very young infants without the requirement of any type of behavioral response. When we start with EEG, multiple signals can be derived from the EEG signal, and I'd like to highlight the work from 3 of these dependent variables. The first is to examine oscillatory power and peak frequency as measures of neural synchrony. As seen in the panel left, the signals can be broken down in a frequency domain with higher power in the lower frequencies, as shown in the power spectrum graph. Each of these frequency bands are thought to be related to different aspects of neural maturation, attention, cognition, and learning. The second is event-related EEG activation, which represents neural responses during stimulation or tasks, and can tell us about the amount and timing of neural processes. As we can see in this graph, these are ERP responses to faces at 6 months, 12 to 30 months, and in adults evoked by the same experiment using the same equipment. The third is connectivity or coherence, which are measures of functional interactions between neural systems. This can provide us information on how processes are related to each other and circuitry formation. Breaking these down, alpha power within the oscillatory frequency area, alpha power represents cortical idling, with suppression of alpha reflecting the onset of attention, cognition, and learning processes. A number of groups in recent publications have highlighted the relation between peak alpha frequency in early development and cognition. In 12-month-old infants at elevated risk for autism spectrum disorders, peak alpha power is related to nonverbal cognition, both in elevated risk groups as well as in low risk groups. This is work that was done by Dr. Leno and combines 3 different data sets that include EEG activation during resting state in infants at familial risk for autism. Peak alpha power is also lower in infants with tubular sclerosis complex compared to typically developing infants, and shows a similar relationship as previously identified to verbal and nonverbal cognition, as you can see in the graphs below. This relationship is also present in toddlers with autism spectrum disorder. In typically developing toddlers in red, peak alpha matures with chronological age. However, in autistic toddlers in blue, this maturational relationship does not exist, and instead, maturation or change in alpha over age tracks with cognitive ability. This might suggest that peak alpha power in infancy and toddlerhood could be used as a marker of cognitive maturation. There are several event-related potential components that inform us of the sensitivity and specificity of social neural processes, specifically those related to processing facial images, like peak alpha, which seems to index a normative developmental process. In infants who develop ASD, we can also see normative processing of faces in relation to we can also test how normative processing of faces develops in response in autism spectrum disorders. And the deviation from this pattern has been found to be related to the level of symptom development in these infants. In our work at 6 months of age, elevated risk infants, those with familiar risk for autism outcomes, show alterations in response to faces at both frontal, depicted here, and posterior, not presented electrodes. In this case, we saw alterations in the sensitivity and specificity of these processes to faces versus comparison to non-face stimuli. You can see the tiny little red dots on the far right of the graph C and F. Those are the responses from the high-risk infants who develop ASD, and you can see just generally the pattern of responses to social and non-social differ greatly in that group compared to the other groups tested. Speed of processing of faces, but not processing a non-face stimuli at 6 months of age, is also related to the severity of autistic symptoms at 24 months of age. In another consortium, the BASIS collaboration in the United Kingdom, led by Mark Johnson and Emily Jones, they also show a similar finding in a larger sample, with 8-month responses to faces compared to a non-face comparison stimulus related to autistic social functioning at 18 months of age. The speed of the N290 is highlighted in the black arrow in graph C. A posterior face-specific component in infants provides good classification accuracy for later ASD outcomes. Graph B from Tai et al. In 2022 examines the use of multiple ERP signals obtained with the same experimental response to faces and non-face stimuli, suggesting that even better predictive potential can be created through the use of multidimensional face-specific biomarkers as well as visual processing general biomarkers. Of interest, this work aligns with the role of face processing and symptom presentation throughout development. Work from our lab demonstrates that in autistic children aged 2 to 5 years of age slowed, processing relative to age-matched peers, processing of faces and differentiate of faces from other stimuli is related to autism symptoms. And Newhouse et al. Has shown that these early signals at 3 years of age to faces are related to ASD and anxiety symptoms at 15 years of age. That is, autistic toddlers with faster processing of faces, more neurotypical response profiles, have less autistic symptoms at 15 years of age and less anxiety as adolescents, above and beyond where their original symptoms were. So it suggests to us that these early building blocks of social processing help to lay the formation of later social skills. Shifting over to connectivity, while not getting into the details of how we measure connectivity, we have seen a number of studies in autistic adults that show that structural and functional long-range connections are altered in individuals with autism, specifically cortical-cortical connections. Atypical developmental trajectories for white matter growth have also been found in all major tracks in autistic toddlers with high autism traits, as well as recent findings in children with ASD. So we take this work and we are trying to replicate it using EEG in, again, our high-risk models. From the Nelson lab, coherence, or the connectivity in the alpha band, which we talked about earlier, specifically intra within hemisphere connection, was found to be decreased in infants at 3 months of age who had elevated autism behaviors at 18 months of age. Of interest, this was during language processing. So the 3-month-old babies were listening to phonemes of their natural language, and the connectivity was measured during those processes. This 3-month-old signal is also related to expressive language at 18 months, again showing that it could be a building, the connectivity or coherence of these signals at 3 months is a building block for later expressive language development. Orakova has, and now recently replicated by Hartson et al. With the basis sample, found that overall resting state connectivity is increased in the alpha band in 14-month-olds, so a slightly different finding, but in older infants. And the pattern of this response is related to the severity of restricted and repetitive behaviors in the autistic sample at 24 months of age. In these two graphs, you can see the areas where the connectivity is stronger, or there's greater connectivity in the high-risk or elevated risk group at 8 months of age who later develop ASD in comparison to the low-risk comparison group. In addition, you can see in the graph A, you can see the relationship in those high-risk infants that develop autism and their repetitive and restrictive behaviors and the extent of their connectivity patterns. In contrast to the work from the UK group, Dickinson from the Justy lab has found that there's reduced inter or between hemispheric connectivity in infants with TSC who later develop autism. While these findings are less consistent than what we see in peak alpha power or in event-related potentials to faces, critical questions still need to be answered about the relationship between structure, white matter development, and function, the functional circuitry under these task demands. This extent of and specificity of the circuit activation may link to behavior, with early over-connectivity resulting in a system that is inflexible and increased in the rigidity of repetitive behaviors and decreased learning, the decreased ability to learn new behaviors. While less connectivity may result from a system that is may result in a system that is more cognitively and developmentally impaired. That is the possibility that the ultimate connectivity profile changes over time and is dependent on the stage of development and their relationship to the learning demands. We want a flexible and fluid brain that can build new connections and stabilize reoccurrent and useful connections. And this is an ongoing process over development as we build and support and get rid of connections that are no longer necessary for the efficient behaviors. Shifting a bit to eye tracking, I will quickly review several studies that suggest altered attention in pre-symptomatic infants from the lab of Dr. Schick. Charwaska and Schick have identified that 6-month-old infants who later develop ASD have decreased attention to complex scenes, people, and their faces. And seen in this first panel is an example of the types of images that are used in this research. Now, these are usually dynamic videos, but they can be static images. But you can see in the 3 images at the top, we have periods of action where the lady is making a sandwich, where she engages in direct speech towards the infant, and then where in the last panel, a toy is actually activated and she shifts her attention in a next activation to there. During these infants at 6 months who have elevated risk for autism, elevated familial risk, and then develop autism, spend less time looking at the scene, the person, and the face in these types of dynamic actions or activities. Excuse me. Further in our work, we've shown just in static images, infants at 6 months who are at elevated risk for autism, who develop autism at 24 months, have decreased attention to faces. That is, they're spending less time looking at faces in comparison when presented sequentially in comparison to other types of stimuli. So it seems like the depth of processing and the amount of engagement of single image of faces is decreased at 6 months of age. Further, Schick has shown that speech, when a face is speaking at an infant, so it includes audio and motion cues, infants who go on to develop, 6-month-old infants who develop autism, as shown in red here, have decreased attention to the face under those conditions of where the face is directly speaking to them. And we think the speech integration, how faces move, and the sounds that are affiliated with that are incredibly important for, again, language formation. Further, McDonald has shown from the Justy lab that at 12 months of age, infants with tuberous sclerosis with autism outcomes, as shown in purple, have decreased higher scores or less behaviors, decreased eye contact, more in disengagement, and decreased social referencing in comparison to tuberous sclerosis infants who are matched on IQ and other features who do not have autistic outcomes. These altered patterns of attention to the face, a building block of social communication, are specific to ASD in all cases. That is, they're not found in high risk or elevated risk samples who have other developmental outcomes, for example, intellectual disability or language deficits. So it really does seem to be specific to the social disruption that we see in early autism spectrum disorder behaviors. So how do we think about this? We have emerging consistency in the field of pre-symptomatic studies with the relationship of peak alpha power and cognitive ability. Thus, these measures of EEG are telling us about the fundamental maturation of the brain and how this supports cognition and learning. In both autistic and populations of children at high risk, as well as those at low risk for developing autism, and in those infants that do have autism outcomes, we see this relationship between peak alpha as a measure of brain maturation and cognition, both verbal and nonverbal learning. This suggests that this fundamental infrastructure should be evaluated potentially as a way of understanding, again, neural disruption and processes. And also could be a way that we could sort of think about measuring cognition and learning capacity for infants and tracking with intervention over time. Further, we see decreased processing and attention to the faces through multiple EEG methods, both the event-related potential findings of peak speed of responses. Not shown were differences in alpha and theta power to faces versus to non-social images, as well as attention as identified through eye tracking methods. Thus, this decreased early processing of faces in the first year seems to be a relatively consistent feature of kids that go on to autism. And unlike peak alpha, seems to really be differentiating the autistic social behaviors that we see that are developing in the second and third year of life from other processes such as cognition and language. Lastly, we see alterations in connectivity, although the direction of effects and the behavioral correlates seem to be more unique, as in we're not seeing a lot of systematic replication, although to be said, this literature is much newer, and often some of the studies don't kind of use the same measures of connectivity or connection, and often don't look at the same outcomes. So it's important that we try to increase the rigor and rigor of that literature and those findings, and try to do more replication work before we can draw some firm conclusions. But generally, I think that the coherence literature can inform us, or coherence measures can inform us of these connectivity and circuitry formations that we think are really critical to more complex behaviors. And so we see that early connectivity is related to, in different studies, both cognition, social behaviors, as well as the restricted and repetitive behaviors. And the extent to which connectivity is increased or decreased, and whether that connectivity is interhemispheric versus intrahemispheric, I think is really critical to consider in relation to the neurological demands and maturational challenges of each age period. And certainly we might expect that some of these measures or these metrics will be unique to certain time points based on the developmental challenges that the infant is undergoing. So as infants are forming these building blocks of language and social attention, we might expect that how they connect with other systems, the frontal cortex, with memory systems, will change again based on the overall task domains and their age of development. We've begun to consider different ways of thinking about attention, and often we think of more or less attention as an amount, sort of a frequency or a percent of time, or maybe a speed of processing. But another way to think about attention is that it might be about attention allocation as well as depth of processing. So we think that this idea of categorizing attention as a depth, that we need both fast and fluent attentional systems, but also ones that are able to correctly process the full extent of the information available, are critically important. And so early in development, particularly during learning periods, you might expect that attention to be prolonged as infants are building the neural structures, those structures that are developing to best process and understand those formations. Those stimuli. And so we think about forming a representation of faces. It takes a while to create a gestalt or holistic sort of average image of the faces that are going to be important in your environment. And so we need multiple exemplars of faces in order to build those sort of fast and fluent systems. And then over time, infants are going to be exposed to more and more faces that likely differ from their family of origin and potentially their community of origin. And those systems have to be flexible enough to account for those new exemplar faces coming in. At the same time, infants need to be able to process what the face is doing. So they need to be able to attend to the facial movement, eye directions, how eyes gaze can tell them about what is going on in the near environment, as well as mouth movements, which align with the auditory input needed to form oral language. Thus, it's important to consider how these disrupted processes in any one of these areas comes together in the whole and can disrupt processes. It may be that some of the heterogeneity we see in autism spectrum disorders is due to differences in how these building blocks are being constructed within the brain, and that some infants may have deficits or greater disability in some aspects of attention or processing, which then causes the circuitry to form in a different way than an infant who may have relative strengths in that area. So one can think about this in terms of the role of object and face processing, right? So an infant that does not attend to information in the environment, neither social nor non-social information, is going to have a system that is devoid of the learning experiences in multiple domains from which to form their experiences. In contrast, an infant that has decreased social attention but increased object attention may develop very circumscribed interests or cognitive knowledge, but may not be able to integrate that well into the social context. There's some work to suggest from our lab as well that these might be markers. How that integrates with sensory sensitivities is incredibly important, as we have found that infants with hyper responses to social information, so increased responses, show better growth in development of social skills and less social disability over time than those infants with hypo or lack of sensory processes or decreased processing of sensory information. So again, thinking about this as an optimal U-shaped developmental trajectory is that we may not want there to be processes that are too much or too little, but need to find the right amount of attention and neural activation for a given task at a given time in development. And it's important to consider that these cascade, right? Because the skills that are learned at 6 and 12 months of age in terms of exposure to faces and to language are going to form the building blocks for joint attention and social referencing, which then build the building blocks for shared play with children their own age, as well as shared interactions with adults. Before ending, I'd like to highlight 2 projects that are ongoing. One is with our group where we're testing the predictive models in multiple risk groups. So this work that Dr. Schick and I have done looking at EEG and eye tracking separately, we are now combining to look at the longitudinal development in infants with elevated risk, both those with familial risk as well as those with medical risk. And this study will allow us to be able to see if these early predictive signals are the same, irregardless of which etiological background might be related to your autism symptoms. In addition, the study is also set up to have both social and non-social comparisons and contrasts, so we can begin to work out a model of whether increased or decreased social attention is partnered with increased non-social attention, which might have a different developmental outcome, as I said, as infants who have decreased overall levels of attention. And this is an example of some of the stimuli that we're using in our research study, where we follow infants from 6 months to 36 months of age. In addition, this project from the Justy Lab for infants of early tuberous sclerosis highlights our need to take our early detection science and influence or be related to early treatment science. So we must develop these in parallel so that we have tested treatments ready to be available as we begin to identify risk markers in earlier and earlier samples. And in the JET study, Dr. Justy has partnered with Dr. Kassery, Nelson, and Teal, and they're looking at the downward extension of the JASPER intervention developed by Dr. Kassery, rolling it out prior to 3 months of age, as well as at a later period between 9 and 12 months of age, and the extent to which this intervention may help to change cortical processes in infants with tuberous sclerosis, as well as the likelihood of an autism outcome in this population. As you remember from the early slide, about 50% of infants with tuberous sclerosis will develop autism. So the question is, what is unique about those infants, and can we help to reset the early learning experiences to decrease the likelihood that these children will also have autism-related disability? I thank you for your attendance and your attention during this task, and I'm happy to take questions.
Okay, thank you very much, Dr. Webb, for a very inspiring talk and also for introducing the field to us. So to start the questions, so obviously I think it's incredibly difficult to take any quantitative measurements from such young children. I do see that there is quite a spectrum, right? So where does the look, going forward, if we want to provide some measurements for diagnosis, do you think the huge variability like we need to do multiple measurements, or like because it's from the subgroups of the ASD, are they like the high performance confounding it, or are the children with other disorders such as disability confounding it? Or I mean, look, going forward, how do you think we can maybe make these things semi-diagnostic or diagnostic? Thank you.
Yeah, that's a very good question. And I think that's one we have right throughout the field and at any age, because children perform differently for many different reasons. And infants are particularly challenging, right? Because a 6-month-old infant can't be reasoned with, or can't to perform in a task. What we do find is that we don't find that later diagnostic outcomes are related to whether or not the child provides data at 6 months of age. The factors that are influencing data loss early on are more related to sort of general infant characteristics of tiredness, fussiness, new experiences and that type of thing, rather than the severity of their later symptoms. So I think that is something that is a little bit different than some of the work in toddlers and early childhood, where we do see that severity of symptom does really impact the child's ability to engage with the tasks we're asking them to do. But I think your question about like the reproducibility of it and its use in diagnostic processes is a really good point. We are at the stage where we need more reproducibility in science generally, right? So while we have converging results, we don't have exact reproducibility of these findings. And to take a measure like EEG or eye tracking and then utilize it in the clinic, we have to have those psychometric values to know how stable it is over time and how related it is to other features. And that just simply does not exist right now in the infant literature and should guide us in our next phase of work. That work is starting to be there's a lot more work in that area in early childhood and older samples right now, but hasn't quite translated to the early infancy period. I think multidimensional measures in EEG and eye tracking give us the ability to combine and get to the subtyping, though, that you're speaking about. Because as we know, currently no one measure has done a good job anywhere in our field of identifying a subtype of children with autism. Genetics doesn't do it. We can have children with very similar genetic mutations and very different outcomes in terms of their, autism and like cognitive ability. So I think we're going to have to really consider that any future direction is really a multimodal combination of measures. And right now, the more measures we have that can kind of converge on neural structure and neural functioning, I think the more we're going to be able to do to sort of understand this first year of life. I think it changes a bit as kids get older and have more complex behavior. So again, this is going to really be an age-dependent sort of look. I think the other thing to think about is I don't think we want rephrasing the question of diagnosis of autism. I think what we really want to do is look at kids kids and families that are elevated risk for poor outcomes, because the types of interventions that we would consider for a 3-month-old or a 6-month-old or 9-month-old are really based on good developmental principles and good attachment-based principles. And I think they're going to have wide-based applications. Right now, the autistic interventions we have are not going to downward extend to that age, right? Just as you can't get the kids to do those type of things. So I think that we do have some maybe grace or some room to think about how do these pre-symptomatic profiles change the underlying learning systems, and that how more specific and guided interventions can kind of add the additional kind of support that the infants are just not able to make use of right now. And I think that focus on more broad-based neurodevelopmental outcomes and risk will really help in the first years of life, with the idea that we can scaffold them to very specific autism interventions as children maybe don't respond or as their symptoms actually really converge on an autism-specific profile. But thank you. That was a complex and very wonderful question.
Thank you. Do our other speakers or peers have questions?
Yes. Thank you, Professor Webb. First of all, I would like to bring my greetings to Professor Fred Schick, who was my postdoc supervisor at Yale. And I have a more technical question. So the results you presented right now, the EEG part and the eye tracking part, they were carried out separately, right? And I would like to ask if we construct this experiment as a multimodal and collect this data at the same time, the EEG data and the eye tracking data, what would you think that will add to the result?
I think, Dr. Wang, thank you. And I do recognize you from previous interactions. So wonderful to have you here today. I think that I think that it's fantastic, right? I think there's two things. One is that I think you can see that where kids pay attention and how that directs brain activity is incredibly important. And we've seen that in work with older individuals. If the way where you're looking at eyes versus mouths, right, is really important for like the speed of the activation of the fusiform gyrus in older individuals. So I think that there's ways that we need to pull those things together. Dr. Schick and I, but one caveat before I get to that is that sometimes the more methodologies we kind of layer on, that they make it harder to actually acquire data, right? So getting our EEG systems and our eye tracking systems to always synchronize perfectly, we get more experimental error, right? So Dr. Schick and I, in our current study, decided not to integrate them because we found that our rates of acquisition were much higher if we did each methodology separate. And that's just unfortunately, right, a methodological limitation we have right now, which can be addressed through better technological development, which I don't do. So I need anyone who wants to do that, please contact me. We'll test all your stuff for you.
I'm doing that.
Wonderful. Yes. Well, perfect. So we have that collaboration already set out. But I think it's going to be critically important, right, to do that. So what we're doing is we've collected a lot of our EEG data and our new stuff with the ability to post hoc go back and code visual attention and behavior. So it's not as elegant as having integrated eye tracking, but it will be able to let us know how these shifts in the child's behavior versus what they're watching or seeing are synchronized and didactically related to each other. There's also some fantastic new work in interpersonal synchrony, where both the experimenter or the parent's EEG and the infant's EEG is being recorded simultaneously. And some work in infancy has suggested that we do see neural synchrony in partners that are attuned to each other. And so I think there's even ways that we can think about, again, our eliciting behaviors as adults, or, the partner in the interaction is driving some of this. And it's not only how the infant responds, but it's also what we're pushing out there that I think we need to do a better job. So most experiments are very standardized, but real life interaction is not. And so we need to move to being able to sort of dynamically think about how an infant in a real life interaction with their parent looks and judges, moves information as well as what that brain activation might be. But so I think it's a great direction we need to go, but technologically it's been challenging. And you'll see that a lot of infant work right now does not have the integration of systems that we could do it just because of it just has proven to be a little bit more burdensome.
Thank you. And nice seeing you.
It's wonderful seeing you too.
Okay. Thank you.
I have one more question. Do I have time to ask a question? Yeah. I enjoy your talk, really. And as I know that there are many labs in the world trying to use the social attention as biomarker for screening autism, including our lab and Dr. Wang's lab. So I have been thinking whether those biomarkers are specific to autism, or they can also predict other disorders like ADHD, SLI, like other developmental disorders. And also the social attention in typical children, typical infants, whether those social attention in typical infants could predict their social behaviors when they're growing older. I would like to...
Yeah, I think it's very interesting. I think it is true. Well, I think we know less about predicting social behaviors in infants right now, right? In neurotypical infants, we do have a lot of findings in like the toddler period and older, where we do see that these face responses are related to social ability as well as social disability. So we often see them related to kind of normative social processes associated by the Vineland scales of adaptive behavior, socialization scale, as well as often to autistic measures of social approach or social communication. So we do see in older kids, we can kind of get this, the typically developing group, or we'll see both. We'll see both patterns. Of course, the autism measures don't have enough of the diversity in the neurotypical population to find relationships. But we do know that in neurotypical individuals, right, face processing and social attention is disrupted in individuals who don't have neurodevelopmental disorders, but go on to develop anxiety, depression, and it also becomes disrupted in those who have later on dementia, Alzheimer's, and those type of things. So I think that what you're hinting at or getting at is that social signals are incredibly important for everyone, right? And I think that they can be disrupted in a number of different conditions and a number of reasons why, and the specificity of them to outcome is going to really change with development. So when they're disrupted very early, right, in this first 2 years of life, we do think that's very specific to autism, but one could imagine a child with really severe attention problems who develops ADHD, right, might, with that attentional problem, develop social attentional issues over time, right? And that we must see the social signals become disrupted as a downstream consequence of those attentional disruptions and the ADHD phenotype. So we do know kids with ADHD sometimes have problems with social relationships. So I think of these things as what we really have is a lot of indices of brain functioning that are just general maturational normative ways the brain develops that we've tried to co-opt or use in autism spectrum disorders as a deviation from a normal process. And I think that's going to be really important in our models to think about those relationships. But I appreciate the comment because it's just very complex, right? The way our brain develops and the way it gets off track. Yeah. It's actually very subtle. And the way that, autism forms, I think it's not a huge, like one day, the behavior is fine and the next day it's not. It is a developmental period that is a really subtle, we think, shifting of attention and social processing in the first year of life. And so it's how we, Ami Klin does a lot of really great work this way, right? It's the trajectory of how these skills are developing that he has found to be critically related to outcomes. And I think that's a really important consideration because it might be the trajectory in the neurotypical group for this versus low risk groups versus the high risk groups, rather than any given moment that also might tell us which children are most at risk. But thank you. Yeah, I'd love to hear more about what you're doing, and it's just fantastic to have so many collaborators around and colleagues around the world doing this work because it's a difficult disorder and needs a lot of attention.
Okay, so thank you very much, Professor Webb, for a wonderful talk, and Dr. Lee from Peking University will introduce the next speaker.
Well, I'll introduce the second speaker of our section. Who is Dr. Wang Quan, who is a professor of signal and information processing at the Xi'an Institute of Optics and Precision Mechanics, Chinese Academy of Science. Wang Quan has been studied in Sun Yat-sen University and Frankfurt University in Germany for a PhD, and she has been working in Yale University Child Study Center as a postdoc and research associate for many, many years, under the supervision of Dr. Freshik and Dr. Kasia Charaska. I personally know Quan for many, many years when she was back in Yale. So her research mainly focuses on biomedical signal and image processing and its application for brain-computer interaction and neurodevelopmental diseases. So including task-based multimodal algorithms and biomarkers development with technologies like eye tracking, EEG, FNIRS, and biologically motivated neural networks. So her title today is Eye Tracking-Based Study on Brain Reward System of Autism Spectrum Disorder. So that's welcome, Dr. Wang.
Thank you. Many thanks to inviting me to this great conference. So my talk today, so everyone sees my slides, all right? My talk today is eye tracking-based study on brain reward system of autism spectrum disorders. So first of all, we all know that our eye is not working as a camera. So how we did eye tracking is because we have foveated vision. So in our vision system, only the fovea, you have a very sharp and high resolution of the picture. So it's like what we see now on the screen. Only the central part within the fovea, we have a very clear and sharp input. That's how we track the eye movement. We track the center of the pupil, and we track the focus of our visual attention. So it starts very early eye tracking experiments. Even in the 1950s, we have saccade and fixation identified very well by a high-speed camera system. And saccade is the highest speed that the human muscle can reach. And usually during saccade, you don't have visual input. And only during fixation, you got visual input from what you see and what's the information from your vision system. And let's start a little bit with the eye tracking history. The first precise and non-invasive eye tracking actually started more than 100 years ago, in 1901. And the first head-mounted portable eye tracker also it's more than 60 years ago. And the real-time human eye tracking interaction, which is very important for human-computer interaction, started in the 1980s. And then in the 1990s and the beginning of the 21st century, we got different kind of eye tracking studies. For example, visual stability across saccades, memory and real-time representations, and gaze translational aids, which is real-time interaction, and also saccadic contingent update in virtual reality. And nowadays, usually when we see eye tracking system, we see two different types of eye trackers. First of all is the head-mounted eye tracking system. That is, as here, which usually it's a head-mounted or a glass-like system, which is more portable. You can walk around in a real-world environment or during driving. The second one is a desktop-mounted system. That is, usually we carry out this type of experiments in the lab. We project everything on a computer screen, and then we do the eye tracking towards the computer screen and track where the kids are looking at. And nowadays, also a lot of commercial, like the technical companies are interested in eye tracking. For example, Google, Oculus, and Apple. And they bring a lot of other techniques, for example, virtual reality, mixed reality, and then which is some new tech we can also apply in our laboratories. And on the other hand, on the other side of the screen, we can see that the eye tracking visualization, usually we can see a heat map. The more visual attention we got at certain spot, the hotter the visualization color becomes. And this is our laboratory setup, and which is the sandwich lady scenario. And first of all, I will bring a very interesting experiment we done in young infants. And this is also 10 years ago, when I was during my PhD years, and we do the infant gaze contingency. First of all, what is gaze contingency? For example, this trial on this picture is looking at the toy. So looking at this toy usually will not bring any outcome. At most, it may trigger a social interaction. So for example, the parent of the child may find out it's looking at the toy and will bring this toy to the child. This is in the real world scenario. However, on the computer screen, we can do a full perception, vision and perception loop. And as we know, eye tracking is more than just observing the world. So eye tracking is important for social behavior. That I say this child looking at this toy may bring interpersonal communication. Eye contact communicates emotion and brings shared attention. And gaze contingency, which is an action perception loop, which started in the 70s and also in 2012 and 2011, there are studies carrying out this gaze contingency with infants. And this is one of these studies. So it's very simple setup. So on the computer screen, we have a red button, which right now is on the right side of the screen. And once the child look at the screen, it triggered by gaze. And in 600 milliseconds, it will trigger animal picture, which will show on the other side of the computer screen. And this picture will disappear after 1.5 seconds, and this button can be triggered again. So our participants are very young infants that are 6 months old and 8 months old, typical developed children. And this is a small video, so let me play it. Okay. So green dot is gaze point. So this is 6 months old, who is looking at the red dot, triggering the pictures and doing anticipatory eye movement, moving to an empty place to anticipate the picture to show up. And he seems very excited. And this is a replay. And we have a slow mode of one triggering behavior. So the child look at the red dot and move his eye and wait for the animal picture. All right. So from this experiment, we can see, first of all, for the number of triggerings, we have more clicks from 8 months old comparing to 6 months old, which shows natural development. And when we look at the average fixation duration or dwell time, we can see that the kids looked more on the pictures, which has more texture, more complicated and visual input details, and comparing to the red dot. So from this comparison, we can also show that it is not trying to, so when the kids, they look at the red dot, they are trying to trigger it. They are not doing observational fixations on the red dot. And on the other hand, we can also see that the kids, they are not only doing responsive reaction towards the pictures, they are also doing predictions. So the reaction time, if we see here, the time 0 is the time the picture show up. And they are this negative average maybe 300 milliseconds, which meaning the kids, they are doing anticipatory eye movement. They move the empty part of the screen and wait for the picture to show up. Of course, the main part of the reaction time shows up at 300 milliseconds after the picture appears. And also we see a training effect so that the kids with the click number increases, the reaction time gets faster. And this is a first experiment with only one dot. And then we design a similar experiment that we have 2 dots, which are from the appearance, they look exactly the same. But one of them, we call it functioning dot. The other one, we call it the non-functioning dot. The functioning dot, which acts the same as the experiment before, we bring animal picture to the middle of the screen. And from this design, we can show that the 6 months old and the 8 months old young infants, they are actually preferring the functioning dot. They are doing more patternized eye movement from the middle of the screen towards the functioning dot and back to the animal picture, comparing to the non-functioning dot. So here, 2 visually exactly the same images were assigned different values. So here, from this experiment, we can first see that the 6 months old and the 8 months old infants, they quickly learn to control their environment using their eyes. so they can discover the gaze contingency very quickly. And they anticipate the consequence of their eye movement, which is the action and change the display on the computer screen. And also we can see the assign different values to the buttons with the same visual appearance. So with the first experiment, we move this gaze contingency to the application in the autism spectrum disorder, the children, their visual attention studies. So this is a 2018 study with Professor Havaska, and we have impaired value learning for faces in preschoolers. And let me refresh you very quickly about this DSM-5 ASD diagnostic characteristics. The first is persistent deficit in social communication and social interaction. And the second is restricted repetitive pattern of behavior, interest, or activities. And third is symptoms must be presented in the early developmental period. And symptoms cause clinical significant impairments in social, occupational, or other important area of current functionings. And the disturbances are not better explained by intellectual ability only. So they are not only developmental delay or global developmental delays. So the motivation of this second study, we call it a reversal value learning. First of all, we know the attentional selection is mediated by the reward system of the brain. So in our visual field or in our environment, there are a lot of things competing for our attention. So what is the most attractive thing in the environment, which is our attention selection, is guided by the appraisal of the object values, meaning that what you found the most important, you gave the most, the longest attention or the fastest reaction to it. So in the visual attention, the object value is defined by the usefulness or the relevance of the information of that the object provides. So current existing studies, we see a lot of animal studies or human behavior studies. Usually is limited selective attention towards social objects. And very few of them are focused on how the initial value was assigned to the object. So first, we hypothesize that the limited attention to faces in ASD may reflect the disruption of value learning in social domain. However, no one shows there are very few, when we carry out these studies, there are very few work about how neutral objects acquire value in ASD, how this process is related to social attention. And one question, for example, one simple question is someone responding to you more important than someone does not? So here, in the value learning, we also can see their long-term value versus short-term value. The long-term value is that the effects of the value learning can endure days past the training stage. And the short-term is in daily situation, the object value may need to learn rapidly and flexibly, meaning that someone's or some object, how they attract the attention may change depending on the environment or the scenario. And with this study, we have social stimuli, and we also have the non-social stimuli. And our hypothesis is that within group, we're expecting the typical developed, for the typical developed children, we're expecting they perform better in the social value learning compared to the non-social value learning. And for the children with ASD, we are expecting they will do better in the non-social value learning compared to the social value learning. And between group, we are expecting for the group comparison is for the social scenario, the typical developed children perform better than the children with the ASD. And for the non-social learning, they may perform pretty equivalently. And this is also a small video. We have this yellow dot shows the gaze contingent, the gaze point. We see there are two fractal images. One can be triggered, meaning that once the gaze point landed on the picture, it will revolve into a cartoon. And this is test. During test, when the two pictures shows together, it's also the high value object will change into a cartoon moving fractals. And the low value object triggers nothing. So this is the non-social condition. We have similarly the social condition. The social condition is one face with smile towards the participant. So this is the high value, we call it the high value face image. So once the child look at the high value face, it will trigger a smile. And once the children look at the the low value face, the picture does not change. So during test, in this reversal value learning, we still have the reinforcer, so they still trigger the smile for the high value image. So the reversal value learning is we have 4 blocks of training and testing. So in block 1, the image A is the high value object. So we have 24 training trials and 8 test trials right after the training. And in the block 2, we have it in the reversal part, the image B is the high value object and image A will become the low value object. And again, 24 training trials and 8 test trials, and which is the same situation for block 3 and block 4. And the object value alternates between the blocks. So our participants have 2 groups: children with ASD and children with typical development. So we have 25 and 27 kids, and we test their Mullen scales and developmental scales and ADOS and with the social effects and RRB and CSS. So these results, we can show that, so first of all, for the high value and low value selection, actually both group in the social and non-social scenario, they both perform better than 0.5, meaning that they are all learned to look at the high value object more than the low value object. And comparing with our hypothesis, we can see that within group, the typical developed children, they perform better in the social reward learning comparing to the non-social reward learning. However, for the ASD group, their performance in the social learning and the non-social learning are similar. And for the between group comparison with the linear mixed model, we have for the non-social scenario, the typical developed children perform similarly with the children with the ASD. However, for the social scenario, we have the typical developed children perform better than the children with the ASD. So the first question is our reward really working? So, okay, the next, maybe I first show the correlation in the social and non-social conditions. So on the x-axis is the learning in the non-social domain. On the y-axis is the learning in the social domain. And what we can see, the left image is for the learning in the children with ASD. They have a significant correlation for their learning in the social versus non-social domain, and which is not the case for the children with typical development. So the children with typical development, they have they are learning in the social domain is higher than the non-social domain, and the learning process are not correlated. So when seeing these results, the first question is we want to ask, is the reward really working? So does the children enjoy the visual feedback? So we look at the positive affective responses, meaning that some of the kids, when we videotape them during the eye tracking experiments, some of the kids, they respond to the smiling face. They smile back to the computer screen when they see the cartoon of the person smiling on the picture. So the percentage of ASD child smile during the social trials is 8.1%. And in the non-social, the fractal cases, it's 5.2. And for the typical child, it's 9.7 versus 4.0. And from this data, it may not be very large amount of data, but what we see here is the percentage of smiling is not significantly different between group or between condition. So meaning that our scenario, the kids with ASD and kids with typical developed, they enjoy the eye tracking experiment to the same extent, at least not significantly different. So in this study, we first see that it suggests a domain-specific impairment in value learning for faces in ASD. And also, children with ASD may employ a shared mechanism, which show as the association between the social learning versus the non-social learning. So they may have a shared mechanism for learning in these two scenarios. And the study provides motivation for investigation into the value learning system in the brain, and it linked to the atypical social attention in ASD. And the gaze contingent, the setup of the gaze contingency in eye tracking paradigm offers a way that is applicable for children with very young age, which can across a long range of chronological or mental ages. So we don't have any verbal instruction or any design that is hard for kids with intellectual issues or young age. But with the first experiment design, we still have questions. So first of all, this is a reversal value learning. How about the stable value learning task? And the reversal value learning, we have this only two groups, the ASD group versus the typical group. How about the children with developmental delay? So we designed experiment 3, which we call stable value learning. And in this experiment, we first have the baseline condition. So we show only the two images on the computer screen, and they have so in the baseline situation, none of them are gaze contingent. So they are just images showing on the screen. And on the second training phase, which is similar as our reversal value learning. So one of the picture we assign as a high value object. So once the gaze point moved towards the object, landed in the picture region, it will trigger. It became a cartoon moving fractal. And after the training process, we came to this choice test phase. So during the choice test phase, at this time, it will not be triggered by the gaze contingency. So it is exactly the same setup as in the baseline. So you see two images displayed on the screen at 4 random positions. So similarly, this is the case for the social scenario. So one phase will assign as a high value phase, which will smile during the training phase when the children look at it. So during this experiment, we invited 3 groups of participants. So children with ASD, children with developmental delay, and children with typical development. And for this 3 groups, the ASD and TD group have they are similar regarding chronological age, but the children with in the DD group, they are older than the other 2 groups. For verbal IQ and non-verbal IQ, we can see the ASD and the DD, they are comparable for the verbal IQ and non-verbal IQ, and both groups are lower than the TD group. And first of all, we look at the mean dwell time, so how long they look at the face and the fractal during baseline. So we can see for the ASD group and the TD group, both of them, they don't have significant differences when looking at the faces or looking at the fractals. However, only the TD group, they spent longer time looking at the faces comparing to the fractal condition during the baseline. And this is the baseline comparing to the choice test, so the before training, after training. So the before training, we have this preferential, the high value preferential percentage is very close to 0.5. So meaning that the kids randomly look at the high value and low value objects without any preference. And after training, we can see for the faces, actually the DD and the TD group, they both have a preference for the high value faces, which is not the case for children with the ASD. However, for children with ASD, they have an enhanced preference for the fractal, which is the non-social stimuli. And again, this we didn't observe in the group of DD and the TD. So we also look at the saccadic reaction time for these three groups. So for the faces, the reaction time are actually the same. However, for the children with the ASD, they responded faster to the non-social scenario. So they react very fast towards the fractal, the high value fractals. And in this experiment, we have found that increased attention to object in preschoolers with ASD, they may be associated with enhanced value learning in the non-social domain. And also I didn't show the visualization, but we also found a higher preference for the high value faces associated with lower severity of autism symptoms from the ADOS2 CSS. And also higher preference for the high value fractals actually associated with higher severity of autism symptoms. And in this study, altering the attentional priorities affected the learning opportunity in the children with ASD, meaning that they are not allocating their resources towards social activities or social objects, given that less opportunity for the children to learn the social domain, anything associated with the social domain, which may also bring a cascade effect as Professor Webb presented in other eye tracking experiments. And as I say, this is a long story. So in a couple of years ago, I reallocated back to China and work at the Chinese Academy of Science, Xi'an Institute of Optics and Precision Mechanics. So right now, we're interested in the we know we are interested in the reward and how this reward pathway is constructed. And we are interested in the posterior superior temporal sulcus and the nuclear accumbens. So from here, we have colleagues working on the optics. So right now, we are very interested in a new technique that it's not new, it's a worldwide, but new to me. So let me bring two slides about optogenetics. So first of all, what is opsins? For people working in the signal processing field, opsin is a light sensor. So it's a protein which forms part of the visual pigment rhodopsin, and it's released by the action of light. So this is two examples of opsins, or we say the two light sensors. First of all, the CHR2. CHR2 is activated by the blue light. So both of them are ion channel proteins. So once you shine blue light at certain wavelengths, it opens the ion channel, and let the ions move into the cell membrane and activate certain behavior. So these are the curve of activation for the CHR2 and for the MPHR. And the MPHR, on the other hand, is a yellow light activated iron channel, and it allows negative ions move into the cell. So both of them, they work as a light sensor. So once you shine certain wavelengths of light, it gets activated. And once it's strong enough, it can activate certain type of neuronal behavior. So this is also from a very classical review paper. So first of all, you get a promoter to drive the expression. And also this can get a specific start of what type of position to start the translation of the protein. And it gets into certain virus as a vehicle, and the vehicle gets delivered to the target animal and gets infected to certain type of cell type. So comparing with the traditional electronic stimuli of the neurons, the optogenetics is very specifically targeting certain type of neurons. So from this genetic process, we can select which type of neuron we are interested, and we can select which brain region to apply this virus infection. And we shine the light through this optic fiber. And once it's triggered, the light sensor, we say the opsin is triggered. It can activate certain type of neuron at certain part of the brain. And comparing with the electronic stimuli, we see it has no side effect of triggering the other neuron type, which is allocated adjacent to the target neurons. So this is a study that work on the SHANK2 mutant mice, which is also a group allocated in Xi'an, and we started collaborating with them and compares the SHANK3 mutant mice with the wild type. So we can see the shape of the synapse. First of all, they are different. And from the behavior of the comparison of the wild type and the knockout, SHANK2 knockout mice, we also see that the wild type actually enjoyed interacting with the stranger mouse in the third chamber, and the knockout type didn't show this kind of social behavior. And they used optogenetics to activate the ACC, the neurons in the ACC, and they found out this can actually, after the CHR2 modified by the optogenetics, this knockout mice show a similar behavior comparing to the wild type, which means they retain the social motivation and started to interact with the stranger mice. And once they turn off the light, means the optogenetics is deactivated, and this behavior disappeared again, and it became like the knockout mice. And there's a second study, which is also a very new study from Dr. Hu's lab, which is last year, 2021. And this shows like when there is a social partner on the other hand of the door, the rat preferred to interact with the social partner, and the poke on the there's a port which can open the gate and allow them to interact with the social partner. And these three figures actually are measuring the level of their dopamine release. And you can see no matter it's a male or female or juvenile mice, all have the rise of dopamine level very significantly. So meaning that this is a pure social behavior, which has nothing to do with mating behavior or other things. And they also tried a different part of the brain, how to, this is the amygdala part of the brain. And once they used optogenetics to light up this part of the brain, and they find out even on the other part of the chamber, there's no social partner. It triggers a very similar type of social behavior, like the mice will poke on the active port, that simulating a social interaction with the the mice on the other side of the chamber. And their results show that this is a pathway from the medium pre-optic area towards the amygdala part of the brain. So from our lab, we just started the optogenetics platform construction. So this couple of years, we still have the eye tracking experiments with EEG with fNIRS. And however, this is our ongoing work. I will stop here. And we are interested in wild collaborations, not only in the clinical part, also in the this different mechanism investigations.
Well, thank you for your talk. And I have several questions from the platform. You could check. The first one is for the smiley face versus non-smiley face experiments. Is smiley face whether the smiley face is replaced with a sad or angry face? What happened?
We are not. I think it's a very interesting question. So first of all, we create smiling is because we want it to be a positive social interaction, right? So it's a stranger because the face is from someone you have never seen before, and we would like to create a reward, and that's why the first selection we make a very positive response. So I think angry face or something, they still triggers a social interaction, but it may not be as rewarding as a smiling face. So yeah, I think it's very interesting, and may want to explore further. It's like how the response. I mean, maybe angry face is better than a non-responding face, right? So at least it's responding to you, but it's less rewarding comparing to a smiling face.
Yeah,
I think it's something worth to look at.
Yeah, I think maybe they're like more sensitive to angry faces, according
Could be.
Yeah.
Could be, yeah. But less rewarding. I still feel it's less rewarding. It's more maybe more valence wise maybe higher.
Yeah, according to my new theory of arousal, like they're more aroused. So another question is that do children with autism have less or more aversion for negative social stimuli? I think there are similar questions like negative social stimuli, like angry face or sad face, or socially threatening stimuli. So what do you think? They are more or less?
I'm sure. I think this is worth further experiments to carry out to see how they respond to that. But it could be there's another possibility is that they are less responsive to the emotional changes. So, for example, children with autism, they're sensitive to the different emotions of the faces. So
Can I ask a question?
Sure.
So you've done experiments with very young children, like 6 to 9 to 8 months, and also like toddlers. So so we would more think that younger is harder, but maybe, I mean, are the 6 to 8 months easier for some experiments because they don't know how to complain?
Well, yeah, that's actually at least for the typical developed children, the toddler age actually are the most difficult for the experiment and demonstration. Have the mobility to moving around to jump out of the chair. Harder to, I mean, sometimes you need to negotiate with them before the experiment, saying that we would like to invite you for a very, very cool experience. And once they say yes, and things usually goes better.
Okay, thank you.
Okay, thanks.
I have a final question. So I'm thinking of the theory of repetitive behaviors. Like some people think the repetitive behaviors are actually rewarding for them. Like, or they're doing something repetitive or seeing something repetitively moving or rewarding to them. But others think repetitive behaviors like could reduce anxiety or comforting that function. So what do you think? I think like some of your data may support the rewarding theory.
Very interesting question. And I think these two hypotheses may not conflicting with each other. So rewarding could be one thing. Comforting could be another type of rewarding as well. So what we are looking at, why I move towards more biological part of the thing is, first of all, a lot of dopamine, dopamine play a huge role in learning and in brain development. So a lot of things could be associated with reward, and even like Parkinson's disease, Parkinson's with the dopamine pathway. And actually, for autism, we may not even restrict it to the dopamine pathway. It could be endorphin or other type of reward. So explore more.
Well, okay. Thank you for Dr. Wang's talk and your answering for those questions.
Thank you.
And so we move to the third speaker of this section, Dr. Li Ming from Duke Kunshan University. Dr. Li Ming received his PhD degree in electrical engineering from University of Southern California. And he's currently an associate professor of electrical and computer engineering and a research scientist in the Data Science Research Center at Duke Kunshan University. And he's also like adjunct professor in the School of Computer Science in Wuhan University. So Dr. Ming Li and I were colleagues in San Yat-sen University many years ago. So we have some collaborations of using machine learning algorithm to help classifying children with autism from typical children. So now his research interests are in the area of audio, speech, and language processing, as well as multimodal behavior and signal analysis and interpretations. As I know, Dr. Min Li has built a multimodal platform to analyze some behavior, signal processing, and behavior data from children with autism to help the autism diagnosis and assessment. I think this is also the topic of his talk today. So let's welcome Dr. Min Li.
Thank you very much, Professor Li, for the introduction. And I'm going to share my screen. Can you see the slides?
Yes.
Okay. Okay, thank you. So I'm very honored to give a presentation, and the title is Multimodal Behavior Signal Processing based assisted ASD diagnosis. So today, mainly I'm going to talk about diagnosis, assisted diagnosis actually. So the outlines is as follows. Number one, I will introduce our early work. Actually collaborated with Dr. E. It's about machine learning side. So once we have collect some eye tracking data with ASD group and TD group, and how can we use some machine learning algorithms to distinguish those two groups and make some early detections. And then our main focus for this talk will be assisted ASD diagnosis using the multimodal behavior signal process. And then we'll introduce a 2 generation of system. We call it Young Hope system, which is generation 1, generation 2. And I will introduce them one by one. And then in terms of how we design the system, how we design the protocol, the paradigm, how can we measure the signals, how are we going to process the signals, eventually interpret the results. So that's basically the focus of today's talk. Okay. So the first, as a previous 2 speakers mentioned, that eye tracking data is very useful in the ASD detection. And then usually people are using some 2B devices to do the eye tracking detections. Of course, nowadays a lot of methods right now do not rely on a third-party devices like 2B. For example, we could directly inference the eye fixation or eye gaze using a camera. However, if you look at the precisions, right? If you want the high precision measures, then you probably need a professional device. But if you are going to measure the eye tracking fixation, eye gaze in a long range or in a wild environment without wearing any wearable devices, then the camera might be a better choice. So I will introduce our early work actually published in 2019 and 16 and 2019. It's basically we're utilizing a data set shared by Professor Ili to us. Essentially is the different child, I think the age is around 4 to 11 years old. And then when they look at a set of face images, I think the set is a total number is 60. They each of them are basically look at a set of images. It's a face images, and then we record the sequence of eye coordinates. And then once you have the sequence of eye coordinates for everyone, and then you can make some machine learning analysis, right? And then we have 29 ASD and 29 TD age matched and 29 TD IQ matched. So the technical question is, how can we distinguish these 2 groups? And then at the time, I think we only have the eye coordinates, right? We don't have other informations. So actually, we've designed a backup words feature presentations. Basically, we're going to divide the images, face images, into different you can say grids, right? And then we're counting or we're accumulating how much time the child is looking at each grid cell, right? And we also use the n-gram language models, mostly like the how likely you're going to jump from this cell to that cell, right? And then with some history where you jump from, and then use n-gram language models to measure the saccade kind of phenomenon, and then use the backup word feature exploitation to represent some kind of fixation informations. And then but in order to do that, you need to design a dictionary. So basically, where you locate each cell, right? So how frequently that each cell get spotted, right? So that's so the core technical problem is how to define the dictionary. And once you've defined the dictionary, in the testing cases, you can just ask a child to visualize the same set of images, and then you can get their eye coordinates generated by 2B, and then you can basically map to the dictionary and get those features, either the static features or dynamic features, and combine them together to make the prediction. So that work actually is effective. Actually, is related to some early works in the psychology. Basically, they are using area of interest to measure the different cells. right? And in the early days, basically, if you look at in the left side, in the early days, the people just manually draw some areas, and they are just counting how much time that the physician actually, or the eye contact spots actually accumulated on each regions, and then they can use regions drawn by hand, or use equally distributed cells and so on. And then we think that we could do a better job by using data-driven methods to draw the different grids. So basically, in 2016, I think we published a paper basically talk about using k-means to automatically generate those cells. Of course, you can see from the figures in the middle, right? From the middles. Right, so that if the eye physician point, right, frequently reach some certain regions, and then of course you will have a denser grid, right, rather than some places that have less data points, and then you will have sparse grid. And then in the second method, basically, when we think about, okay, so the k-means is a kind of unsupervised clustering method, basically does not take the discriminative information as a consideration. Right here, the method 2, we think about want to allocate more dense cells to those regions that are discriminative in terms of TD and ASC, rather than we spend a lot of cells on some regions that are not informative. So here is basically we're using main shift methods to design a better dictionary. So if you look at the left column, right, on the top, it's basically AOI, the traditional method. And then here we make a prediction about 29 ASD versus the other 58 TD group. And then you can see that the accuracy, right, actually get increased if you use the k-means clustering, and then if you use the supervised mean shift method to better allocate the cells, you can get better ones. So actually, this is our early work, just shows that once we have the eye tracking data, of course, I think there are tons of different paradigms you can design to serve as potential biomarkers. And then when you collect the data, right, how do you utilize the data to make a classifier that can be used in the assisted diagnosis? And then we basically just show our early works that how you analyze the eye coding, eye tracking coordinates to make a prediction. So the main focus of today's talk is basically about the multimodal behavior signal processing, and I'll introduce the Young Hope system. So the Young Hope system 1.0 was designed in 2015, I guess, and then we started to establish the system and collect data and do some analysis, and then get the results, I think, until 2018, around at the end of 2018. And then from the Young Hope 2.0 system actually started from 2019, and this year got released, so that we're going to show you the historical experiences by designing this kind of systems. So 1.0 system actually was designed in 2015. That moment, we just used some HD cameras. We have 8 HD cameras, and then we have one old version of Kinect. And then you can see that there are table here, and then our doctor, parent, and child sit right. It's just the traditional setup when people are doing adults, people doing other kinds of diagnoses, and then we're just silently watching about what they are doing, right? And calculate all the data simultaneously. So this is what we do in the 1.0 system, okay? And then the system actually is hosted in the third affiliate hospital of St. University. Unfortunately, right now, since the second 2.0 system is ready, so this system is already not used. For this system, actually, at that time, we actually designed the 3 tasks, which is very important to perform early diagnosis. It's essentially they are response to name, a separation union, and response to non-social stimuli. And that are the 3 paradigms that design in the 1.0 system. And then we're going to have a label at 2 levels. The first level is basically we directly have a professional doctor to label the score of each paradigm. For example, response to name, they may have 0 score, score 0, 1, 2, and so on. And then we're going to have a paradigm level labels, as well as a subject level labels in terms of ASD or TD. Or actually, you can see that we also have a development delay. Actually, there are 3 groups. And our input data are 8 cameras. They are synchronized collected, and audio data as well, and 1 Kinect data for capture the depth information. And our participants are 111 children. Okay, their average ages is around 24 months. And the environment is basically lab-controlled scenario. Okay, these are the numbers for ASD, TD, and TD. So at that moment, we have designed the 3 paradigms, right? So basically, number 1, response to name. This is quite common, right? So basically, the doctor is calling the child's name at the position it was 135 degree, right, away. If you look at the child, it's child center, and then the child needs to turn the head off, right, towards the back direction to look at the doctor. And then of course, you will see some clearly response or some no response. There were different scores. And then they also have designed a paradigm called separation reunion. Essentially, the parent is going to leave in front of the child to the door, and then we're going to observe how the behavior of the child looks like. The child may chasing the parent to leave, or the child has no response, right? Something like that. And if you look at maybe I can show some videos, I guess. Right. So in this case, that the child would chase the chase the parent to move and so on. Right. Okay. And the third task is to respond to non-social sound stimuli. It's basically we have a small toy. The toy can make some sound. The sound actually are controlled by a controller. The controller are in the pocket of the doctor. So the doctor use their hand in the pocket to trigger the toy to make some sound. And then we'll look at whether the child will sound, and the toy at the back of the child will attract the attention of the child. And then whether the child will point to it, will look at it, will do some visual reference with the parents or the doctors and so on. Right. Those are the design for the non-social sound stimuli. Okay, so how about the first generation system, the data processing? Actually, the first generation system is more like a task driven, so that the doctor designs a certain task, and then we collect some data, and then once we have the data, the data sets, then you start to analyze everything offline. Right. So basically, you do the detection of the pose, detection of the face, of landmarks, the gestures, the audio content, and so on. Right. However, as I will show you some limitations of this kind of design, but you can still, based on the cameras, there are 8 cameras. No matter where they look at, there's supposed to be a camera that can capture the frontal face. And then based on those multiple cameras, you can capture their behavior, the visual, the vision-wise, the behaviors, right? And you can use a set of speech modules like speech recognition, speaker recognition, speech dilatation, and so on. You can try to understand when, who is talking, and what is the content, and so on. And based on those audio and visual information, then we can make some predictions. So the predictions is on the paradigm level first. For example, this is a response to name, right? So once they have input videos, right, we can automatically chop it to core number 1, number 2, number 3. How do you chop it? You actually chop it based on the speech recognition results, right? For example, here, core number 1, number 2, number 3. Once you have detect the call, your name call is based on the speech recognition result. You can find they are calling their child's name. And then immediately you are using the visual cameras to look at where the child is and whether the child will turns the head to watch or pay some attention to the name caller, and then we're going to look at their face and their turning angles, basically calculating some angles based on the landmarks, and they're also going to verify who they are looking at, whether it's a doctor or parent, and based on facial recognition modules. And then eventually we can get some not only the score 102, but also we can calculate some latency or latent features. For example, latency, intense, durations, and head pose, and so on. And I mentioned later, I will show you later that those detailed quantitative behavioral features will also be complementary to the score features that are designed before. And if you look at the response to name task itself, right? If you look at the task itself, and then here the if the prediction is just the label just 0 and 1, whether the child basically have a response or not, and then you can get very high accuracy. But the high accuracy is not, I will explain later that this is just a binary classification, which is not very informative, right? If you increase the number of the classes from binary to 6 or 7, which is the second version of the system, that we have more detailed parsing about the behavior. We're going to have 6 or 7 different levels to describe the paradigms, and then the accuracy per paradigm will drop. However, the information actually delivered will be more rich. Okay, so you can see that by current machine learning methods for the behavioral signal method, we can get fairly good accuracy for detecting the response to name, the behaviors. And similarly, we can also do the supplementary union, and then the techniques we're going to use is for person re-ID. The person re-ID is kind of a technique that in the security application, for example, in a city, there are tons of cameras, right? And there's a suspect show up in one camera, and then you're going to use the rest of the camera to search where the suspect show up again. And here, basically, we have 8 cameras. We can use multi-view information to track where each subject, their trajectory is so on. And then based on the video-based person re-ID information, right? And then we can understand their distance. We can understand where they are, and by calculating the distance, we can know where when the parent actually going to leave and how the child will respond. But there are some limitations in this version of the system. We only look at the distance between the child and the parent. We did not pay attention to the child's eye contact, the child's child's verbal things, so that will be introduced in the second person system. So essentially, for this paradigm-wise predictions, we can also get fairly good accuracy because it's just binary classifications about this separation reunion, right? And similarly, for the response to non-social sound stimuli, and then we actually chop the video into several clips, and then for each clips, we're going to extract their skeleton based on their behavior. We're going to extract their binary skeleton map, and then based on this skeleton videos, right, we can design a system to directly modeling their behavior. And for this response to non-social sound stimuli, we can also get 81% accuracy for this labeling this paradigm. And then we're actually going to combine those 3 paradigms together. If you look at the figure on the right, here, basically, we have the 3 paradigms. We have middle level features, right? We have their middle level features. And then we have their clinical score, which is our predicted. Here, this is our predicted clinical score, which is every paradigm have 0, 1, or 2, or something like that. And then combining with features so we can make predictions. And here you can look at the input features have not only the score predicting subtasks, but also many, as I mentioned, quantitative features. And those quantitative features could be also very helpful. And if you use them all to make a prediction of AST and TD with the 111 participants around 24 months, and then you can get an accuracy about each one is around 70 something, and then combine them can give you 81%. And this is the first level version 1 system about 81.1. And we have to notice that if we combine the behavior clues, right, with the M-chart questionnaires, we can actually get a much better result, which means actually we did some feature selections to see what happened. Actually, we can, if you combine the model, we can combine the 3 paradigm with M-chart. We only use 3 questions from M-chart by feature selection. We only use 3 questions from M-chart, and then we can get 89. What are those 3 questions? Actually, number 1, I think one question is about the smelling, right? Whether some smelling issues. Of course, our paradigm does not touch anything about smelling. And another one is whether you have some active or initiative of drawing attention or something like that. We also does not have that paradigm and something like that. So that this will motivate us to enrich the paradigm in the second version of the system. So there's some limitations for the version 1 system. As I mentioned, that it's a lab setup. First of all, lab setup. So it's not standardized at all. And then why we need a standardized equipment? Because let's think about the MRI and think about if you want to use tons of, for example, thousands of, 10,000s of data to do some behavioral signal processing. And then the data collected from one center or one environment or one lab is not enough. We want to collect, we want to merge the data from many labs together, right? But in that case, that the device should be the standard. Otherwise, you can't merge data, right? Different cameras, different views, different resolutions, different setups. It's difficult to merge. So that we want to use a standardized device. And also, since the camera locations, I can tell you joy, right? So every night, the cleaning staff members will lift the cameras and then do some cleaning of the ground and put the camera back. And the next morning when we do the calculation, everything screwed up, right? We have to recalibrate all the cameras. So we want to make the cameras actually the fixed in the wall or some places. But we also need to consider the size of the rooms, right? And also look at stimuli. So right now in this experiment, stimuli are many human generated stimuli. And they want to make the stimuli objective, right? We want the computer to generate those stimuli. And also we want the environment to be small, compact, and really easy to duplicate and use in other places. And also we want to make it a platform that can, for the doctors, very easily to add a new paradigms. We want them just spend 1 hour, they can add a new paradigms and get the result instantly, right? By after they get the paradigms and then after doing experiment, and then they can get a result, get a report in time, for example, in 1 hour, 2 hours, rather than they collect tons of data, give the data to a professor in CS, and the professor take 1 month and get the results back to them. It will not be efficient. We want them, the doctors, to be creative by themselves. They can make a new paradigm by themselves, and then they can get the result immediately, right? So as I mentioned, that the current system is not automatic because you have to manually segment the video into different paradigms. They are not automatic. And also there's some long delays, not online processing. So that actually triggers our second version system, which is we call it Young Hope 2.0. And the design is a standardized and configurable platform and programmable platform for gathering and analyzing the behavior data. So look at this is a cube. We can call it a studio, and this is a standalone device. It's actually a studio where we have 8 4K cameras. And we also have 4 RGBD camera with depth information. And then every camera is hosted on the wall and with some little hole actually to give the lens a chance to see the scene. And then we have a hidden microphone was mounted on the other side of the table. Okay, so this is a setup, and we have actually four projectors. If you look at the ceiling, right, there are 4 projectors, and those 4 projectors can be created in an array, right? They can actually create a unified thing. So basically, the bird can fly from the wall 1 to wall 2, wall 3 to wall 4, right? So basically, the different walls, they are showing different. They're showing one part of the whole image, right? Basically, they can move freely, and then the visualization actually are controlled by a PPT, just like what we're doing, like using a point. So that the doctors can very easily to add a new animations, add a new virtual background to the PPT, and then they can just full screen to show up the PPT, and then everything will be projected on the wall. So that the stimuli would be programmable and it would be all objective. And all the cameras will basically not customized design to detect a certain time, a certain region, or certain people. It actually cover all. We're going to classify, we call it behavior transcription. I'm going to show you later. We're going to classify the behavior information for everyone in this room for every second, so that we can easily utilizing the result to come up with some result analysis. And here is some real device interior looking, right? And we have designed in this 2.0 sequence 6 paradigms. If you look at their response name, social smile, indicating gesture, responding to joint attention, initializing to joint attention, separation, anxiety. And here the paradigm has much more detailed levels of observations. For example, here they have a score from 0 to 5. Some of them score from 0 to 6, 0 to 7. So there are many different levels to quantify the behaviors, and we actually have more power to get the detailed visual the behavior pattern. For example, we can quantify their talk, their speak, their smile, their eye contact. Eye contact means you have to every second be able to know where the child is looking at. You also need to know every second where the doctor is looking at, and if they are looking at each other, then means there is eye contact. And similarly, we have a pointing, we have a hand gesture, we have pointing, we have visual references, and the other and the trajectories and so on. So the idea is we have all the input data, multimodal data into the system called audiovisual signals. And then the paradigm design could be controlled by the doctors. And then we have multimodal behavior analysis framework. We can generate all those patterns as I mentioned before. And then we're going to use the patterns to, according to the paradigm setup definition, we can make some scores, and then eventually we can do the assisted diagnosis. So if you look at the multimodal behavior analysis framework, actually there are 3 important modules. There are behavior transcription, there are response parser, there are rule-based scoring. So behavior transcription is more like this part, right? Once you have the input of data, we have the camera data, we have depth data, we have audio data, and then we have a bunch of locally hosted behavioral signal processing modules. So why they have to be locally hosted? Because we don't want the signals or data be transmitted to the internet. Everything is calculated local, so that we can respect the privacy, and all the data is stored locally, so that we don't worry about the data get leaked to some internet services. So that all those modules are locally computed. So we have their precision, the gaze, the hand, the body, the motion, emotion, gesture, language, and so on. So once we have those behavior signal processing or machine learning modules, then we call it dialogue. We can have a transcription. We can have the behavior transcription and give to the doctor. Say we know we know the information about the look, point, response, speak, smile, eye contact, social reference, so on. And then we're going to integrate those information, and according to definition of each paradigm, so we're going to make some classifiers. So here is one example, right? If you look at the right upper corner, that actually shows the 3D spaces about where the child, doctor, and the parent, where they are, and then where is their head orientations, and then the black arrow shows where is their eye gaze. And then we also have the bounding box for the behaviors, the pose, and on the right lower corner, we have their face we have extracted the face real time. We have their ID. We have their emotion. We have their other information as well. So that oops, I need to change. Right, so that is all real-time calculations, so that you can get those once you have the videos, right? You can. And then based on that information, we can clearly get the dialogue. For example, the ID, time, where's the funny box, where's the body, where's the face, where the key points, where the hand gesture, where the gaze, and so on. We'll have this files and make a structure. And also we can visualizing the process of each paradigm. So previously the data, right? So if you want to review a paradigm's audiovisual, they have to watch it, right, manually. So right now the doctor can just look at this picture. If you look at it here at the A1, right, at the A1 region, right, it's basically where is the tree? There's a doctor asking those questions. And then to ask the questions here, the gray the contour will show up past the threshold, show you, okay, here's the time the doctor asked a question. And then the blue contour basically shows look at the object, whether the child is looking up. Where's the object? Object is generated on the wall by this tree. And then because the object is generated by the computer themselves, so the computer automatically knows where is the tree. And then they just need to look at whether the child looking at certain angles, and then they can identify whether you're looking at this object. Okay, object position will get changed, and the computer will dynamically change the object location as well, because the stimuli is generated by the computer itself. And then we have the point to the subject, the contour, and we have the ratio references. And then the doctor will know, okay, in R1 region, which is the 2.5 to 3.5 seconds, and then the child is looking at that object, and from the 6.5 to 7.5 seconds, the basic child is pointing to the object, and then in the 10.5 to 11.5 seconds, actually, child has the visual reference with the doctors and so on. So basically, by one images, right, by one figure, you can basically visualize the entire process of the paradigms. And our experiments are conducted like as follows, right? We have collected 95 subjects, and then average age is also around 24 months. And then we have ASD 71, TD 24. Why we have less TD is because these data are collected on the real clinic. So basically, we did not go to some kindergartens ask kids to come here to collect. Our data all collect in the clinic. So basically, when the child go to the clinic, they already have some issues. But after the diagnosis of the doctors, right, they will have a TD and ASD. They will have that's the reason that the TD is relatively lower number of TD, and ASD has the majority of people. And if you look at age description and gender description is also the male as a much higher number of male compared to female, which is the same in the real clinic statistics. And then once we have test children here, we independently go through the doctors to make a diagnosis and go to our system. And our system will have the computer and protocol scoring and assisted diagnosis results, as well as we will have another annotators that just look at the videos, and then they're going to score on each protocol. They are going to score on the subject, so do a calculation as a reference as well for the protocol-wise classification. So this is the results. If you look at the protocol, they are corresponding to 6 paradigms. And then their score level is much more rich. There are 3 to 7. So basically, respond to name, there are 0 to 6. There are 7 levels. So that although the accuracy is relatively small, but if you look at the MAE, mean average error, it's only 0.6 compared to 0 to 6. The scale, I think, is acceptable. And then if you look at computer generated results, it's similar to the So annotators except for response name. But if you look at the MAE, is also acceptable. And then if you look at the consistency of two human annotators, the human annotators accuracy is also not 100%. It's also around the 70 to 80 percentage range, which is similar to our computer's result. And this is at per paradigm, how accurate it is for the computer to classify the score in the range of 0 to 6, 0 to 5, 0 to 4, and so on. And then we also combine those predicted paradigm scores to make a prediction of whether the child is ASD or not. And then if you only use the predict scores, and then the accuracy is not very good, it's only 84%. If you compare to but if you look at the annotators, if you look at the human annotators, their predicted scores is also not very good as well. And then we actually think about, as usual, we can introduce some quantitative indicators. So basically, when we do different paradigms, we want to look at not only the discrete like a scoring measurement, scoring rubrics, but also want to have some soft features like latency, duration, intensity. And then when we use a set of these soft features, and then we make the prediction and accuracy is much better. If you look at the features, this is scores. If we combine them together, we can get right now you can get 88% accuracy. If you look at sensitivity and the spectivity, I think they are okay. This is the confusion matrix. So, I'm not so sure whether I have some time to show a video, but I will stop here and take some questions. So, I think probably I can leave some time for a QA. But in a summary, I think if you want to make a assisted diagnosis system, there's one key information that we need to make the system rather easy to use, and then we need to make the system or standing alone device that the doctor can operate themselves on the device, and they can generate the result in a few hours without any engineer in the loop. Right, so that this can be a real assisted devices, and also that we need to pay attention that we need to make the system applicable to most of the child, which means that if there's 100 child go to this device, go to the system, we want them almost 100% the child can get a report, right? So of course this is why we want to choose behaviors to do as the input, because behavior, audiovisual, non-contact, non-invasive audiovisual data would be much easier to collect rather than other mechanisms, right? At the end, I want to thank our collaborators, Third Affiliate Hospital Science University. Really appreciate their support in the last many years. Yeah, thank you very much, Dr. Yi.
Oh, thank you very much for your talk. And there's a question from the platform. Could you see that in the comments?
All right.
It's in Chinese, like.
All right. The asked question says the current ASD detection and assisted diagnosis devices are using the facial language. All right, so that's a good question. I think it's not strict. I think the eye tracking data could also be a very good choice, and so technically the MRI data could also and MRI data also have very good potential to be used in the diagnosis. But the problem is it's relatively difficult to capture the data. So if we want to utilize the mechanism in real life, we want the convert ratio to be as high as possible, which means that if there are hundreds child go to a lab, you hope that 95 of them can finish the experiment so that you can get a report to 95% people, rather than you only get 5 people successfully finish the experiment and you get very high accuracy for the 5 people, but how about the rest of the 95? So I think non-invasive, easy to capture signals, and then have clearly a theory of why you want to do that, and then the result is interpretable by the doctors. Basically, the doctors will believe, okay, if I don't trust anything about the quantitative feature, just rely on the scores, and I can still utilizing the scores you generated to integrate in my own assessment. That could also be a contribution, because doctor does not want a binary classification yes or no. They don't trust at all. They want you to assist them, to give them a set of metrics, and then they're going to digest it, and they're going to use it in their own way, just like the visualization, the whole paradigm into one figure. And then this will save a lot of time for the doctor. They just spend maybe 30 seconds to visualize the figures, and they understand what's going on in this paradigm. Yes.
I have a question. Sarah has a question. Let's answer Sarah's question first. For those children, they are not classified as autistic by system, but they are diagnosed by the clinicians. Did they share features or behaviors?
Thank you very much. So actually, in the paper, we have a detailed analysis. Actually, see, there are 5 or 6 people, I can't remember. There are 5 or 6 child actually are diagnosed, are detected as ASD by the system. However, there are TD diagnosis by the doctors, and the reason, when we look at it, interestingly, there are a lot of them are the age is more than 24 years old. 24 months old. Sorry. So the idea is some kids when they grow older, right? So that their range of the dynamic movement actually will be more than the child who is 18 months old. So that sometimes the computer will misclassify their behavior just because their movement scale is much larger, so that it will trigger some system to misclassify them as some certain things. Similarly, for the very young children, because even though they are typical developed, but however, since they are so young, then their body size is small, then their movement is also small scale. Sometimes difficult to get classified. For example, they use very tiny finger to point something, and then the system may not be accurately classified that very tiny finger pointing action, so that may trigger some errors. So that might be a reason, but of course, there's a lot of limitation in our system. We hope to further improve it. Thank you, Dr. Swett.
Thank you. I have one final question. Do those children's behavior altered when they realize that they're being watched by the cameras, or they realize that they're watched by someone in the other room using the cameras?
I think that why we want to build a studio, because a studio can render some natural things. So that they may, if you look at the picture, they're just like a real clinic, and because the full walls will be display some images. Sometimes you can change the images to a forest. Sometimes you can change it to a beach, right? So basically, they'll make them calm down, right? It's and then the camera are located at the back of the studio, so that if you look at the wall, you won't easily identify where the camera is. So basically, it's like you hide the camera into some corners where it's not easy to get notified.
Okay, thank you very much for your talk, and I'm gonna ask Dr. Yu Xiang for introducing the last speakers of our session.
Thank you very much.
Thank you.
Thank you very much. So the last speaker of our session is Professor Wang Zhen from the Department of Psychiatry and Cognitive Sciences, and also the McGovern Institute. at Peking University. So Professor Wang Zhen has a background in engineering and medical physics. He started his lab in 2011 at the Institute of Neuroscience in the Chinese Academy of Sciences, both as PI and director of the brain imaging facility. So his lab focuses on integrating advanced functional imaging methods with network computation and neuromodulation techniques to investigate the mechanisms underlying neuropsychiatric disorders, including autism, but also addiction and obsessive compulsive disorders. And another important feature of his lab is that he studies both human patients and uses monkey models of disease where we can use more invasive methods. And so I believe he will talk about his work on how to establish quantitative diagnosis for psychiatric disorders using functional imaging and combined with other methods. So please welcome Professor Wang Zhen.
Thanks very much, Xiang, for this kind of introduction. And I will be the last speaker during the morning session. Okay, as Dr. Yu just introduced, I will talk about the building a Bayesian model for quantitative diagnosis of psychiatric disorder based on the brain imaging data. First of all, let me share my slides. Okay, again, as in a number of talks in this morning, and all the speakers have been mentioned about diagnosis in psychiatric disorder, specifically on autism. And I think that in the audience, all of you may be aware the current diagnostic critical manual is based on the DSM-5 or ICD-11. Those two diagnostic manual is like a questionnaire-based evaluation. It really depends on the physicians or clinical doctors' background or expertise. But here, I want to specifically mention about the imaging, specifically is the brain imaging, not the video imaging, just introduced by Professor Mindy. The brain imaging based diagnosis in psychiatric disorder is also very challenging. Here, I'll give you an example. There is a severe female patient with eating disorder. You can look at the picture. This female, about age 20s, and her height is close to 170 centimeter, but only got weight less than 34 kilograms. If you look at the T1 weighted MRI scan of her brain with your naked eyes, I believe that most of experienced radiologists cannot make a diagnosis based on their T1 or T2 structure scan. Because it's really, of course, right now, we know there is subtle structural abnormalities in these MRI images, but however, it cannot be detected by naked eyes. But in the current situation, we're facing a rapid increased prevalence of psychiatric disorder. Compared to the U.S. prevalence rate about 26.2 in the Americans adults, and now the Chinese society also experienced a fast increase in the psychiatric the prevalence rate of psychiatric disorders for all of them. So the prevalence rate is about 1.1% from 1982 to the current data shows the 16.6% in 2013. Among of them, the number one disease is anxiety-related disorder. It's about 7.6%. The second is a mood disorder, 7.4%. And if you're interested in addiction or substance abuse disorder, it's about 4.7%. Of course, those numbers, for example, the anxiety disorder and the mood disorder, they added up together close to 15%. Why is that? Because the total rate is 16.6, right? So, of course, there's a huge comorbidity or heterogeneity. Here, give an example about the autism, since today's topic is about autistic spectrum disorder. We all know that there are very broad range of dimensional phenotypes in autism. Regarding this clinical diversity, there are a number of prominent deficits people can observe from autistic children. For example, the social interaction deficits, the inattention, the hyperactivity, and the sleeping problems, even sometimes comorbidity with epilepsy. All this, not only the clinical diversity, also people right now have found out a huge genetic diversity as well. They're saying probably around over 100 or to 150 genes related to autism, and it may cause the downstream gene mutations of about up to 1,000. So that's the huge clinical heterogeneity and the comorbidities in psychiatric disorder. Actually, not only in autism, but also we know here the comorbidities present in autism with like schizophrenia, ADHD, or OCD. So now the imaging-based diagnosis or evaluation has been challenged a lot because we know that the imaging scan, especially MRI scan, is mostly based on the phenomenon. So the doctors or radiologists that try to make an inference based on the imaging they observe. But we're now facing the three challenges. First of all, the very low accuracy of diagnosis rate based on the MRI scans for psychiatric disorder. We're talking about the 50 or 60% of accuracy. And the second of all, most of the imaging scans or imaging results are lack of specificity or lack of clear biological underpinnings, which means we cannot specifically link the phenomenon we observe from the imaging to specific genetic or molecular explanations for this observation. And the third, most of the imaging results lack of causality, which really hard to make these imaging findings interpretable in the clinical situations. So how can we solve these challenges or these problems? From my own perspective, I think since the imaging can be considered an intermediate phenotype, so we most likely to build a bridge between the genetic causes and the behavior phenotypes. So the brain circuit is kind of in the middle as a bridge to link both ends. So if we really like to tracing back from the imaging observations or the imaging phenomena and find out their more specific biology underpinnings or genetic underpinnings here. Currently, I think in the field there are two major lines of efforts try to solve this kind of problem. Of course, the solution one, if we believe a reductionist perspective or approach, and people can build or people can create the animal models especially here with a single gene manipulation, and the people can create a rodent model or create a monkey model to mimic one or two dimensions of psychiatric disorder. Here, since my lab mainly working on the non-human primates, here I only talking about the non-human primates models here. But the one thing I want to emphasize is I quote a sentence from this perspective paper published on Nature Neuroscience in 2016. In this quote, we should not expect the monkey schizophrenia or monkey autism to resemble the human disorders in every respect. The best outcome will be a behavior phenotype with some discernible similarity to the human symptoms, reflecting dysfunction in brain circuits that are homologous to those affected in a corresponding human disease. Why we're saying that? Because we hope an animal model can be a simplified version of a human psychiatric disorder. That a best or a better model cannot be, cannot resemble the human disorder in every respect, because it's really a complex situation in human scenario. We really want to simplify the human scenario in an animal model with an animal model. So, if we consider an animal model as a valid model, and people usually to evaluate their face validity, construct validity, and predictive validity. And here, in non-human primate model, if we would consider as a valid model, we should expect that this model can be helpful to the human diagnosis or to the human disease intervention. And here, I'll give you two examples. Our lab recently worked on these two genetically modified non-human primate model. There's two genes. One gene is called MECP2, another gene called AMP2. Unfortunately, these two genes are both related to neurodevelopmental disorders, specifically like autism. And in the first genetic monkey model, this Dr. Chang Sun and Zilong Qiu in Institute of Neuroscience of CIS, they both work together to create this autism-like transgenic monkeys overexpressing MECP2. And the second model, later I go back to come back to this MECP2 overexpression monkey. The second model is called the AMP2 lockup monkeys, but it's unfortunately even we observed too obvious the dramatic volume loss, brain volume loss in those transgenic monkeys, from early birth to even we track to 1 year and 2 years old. However, we did not observe the prominent behavior deficits or the prominent behavior phenotypes in those monkeys. So we tried to integrate multiple disciplinary techniques to investigate the circuit mapping from monkey brain to the human brains, and this including image technique, genetic technique, and behavior technique, and the neuromodulation technique. And first of all, we use the genetic technique. When we investigate the MECP2 overexpression monkeys, we are wondering how many or how other genes are altered during the MECP2 overexpression. So we try to collect more data, especially transcriptome data from the macaque neurocortex, and we using this gene co-expression network methods, try to identify the other genes related to MECP2 in this data. we find out that when the MECP2 is changing, the closer related to the like the GAD1, SLC32A1, and this GAD1 is clearly related to the GABA signaling pathway. And when we try to get the primary data from this public data sets, it's actually from NIH Blueprint, NIH it's Non-Human Primates Atlas. From this website, you can download data from about 182 samples from 10 monkey brain regions. However, it's not complete. So right now, we managed to collect close to 1,000 RNA sequencing samples from 111 brain regions. And also, we collected MRI data from 162 monkeys, especially cynomolgus macaques. And we try to build up this transcriptome and MRI data set for the cynos monkey and macaques. And we really like to construct this resources platform to compare with the human brain atlas. And here I can give you an example. The left-hand side is the transcriptome atlas of the adult human brain, and the right-hand side we managed to build this transcriptome atlas of cynomolgus macaque brains. We have just submitted this paper currently under review. So we switched to the behavior method approach to evaluate the MECP2 monkeys, especially their behavior phenotypes. We observe when we raise those monkeys in their cages, we observe prominent repetitive behavior patterns. Here shown in this video, you can observe the monkeys spend a lot of time of doing circular routing inside their home cage. It's kind of mimic the human repetitive behavior paradigm patterns. Of course, we also design a paradigm. It's called the peer separation paradigm. So we build a very large cage. This cage has like half room size here. So this cage has been divided 2 compartments. And we put the MECP2 monkey in one compartment and for 20 minutes like adaptation. And then we put a partner to interact with monkeys. So in the middle, there's transparent window. The 2 monkeys can see each other through this window. And then to see their hard interaction. After 40 minutes interaction, and we remove this partner. So this video just shows immediately after we remove his partner, this transgenic monkey show, you can see here. Yeah, once we remove his partner, and this monkey just started like a circular routing inside the cage. And this behavior is very persistent. If we you look at this video very long, for example, like 20 minutes or even 40 minutes, and this monkey is continuously doing this for over time. Okay. So since we observe a very prominent behavior deficits, that's called behavior inflexibility, we wonder those monkeys either suffer the cognitive flexibility. So we adopt a reversal learning paradigm. It's quite simple. This paradigm includes color discrimination and reversal learning. So the first phase, a monkey trained to do the hand fixation. So they just touch screen and there's a red dot in the middle, and the monkey just touch the dot and to start the trial. And then there are 2 colors presented at the same time. So a monkey have to pick out the right color for the juice reward. So initially, the juice reward given to for example, to the green, and then after a random number of trials, the juice reward will be given to the blue color. So during this switch, there's no cue for monkeys. Monkey have to figure out by trying and error. So we just we use this paradigm to test the cognitive flexibility in these transgenic monkeys. Here I show you the video. Okay. So the touch screen and the monkey pick up the green color for the juice reward. So pick up the green one. It did the right job. It did good job, and it kept moving its mouse because there are mouthpiece inside the mouse and deliver the juice reward. Okay, so it almost 100% correct. And now we switch the juice reward to the blue reward. Okay, so you see he pick up the green one. Oh, now it's blue one. Okay, a blue one, a green one. Okay, when he chose the green one, there is no juice reward, and then why his cat turning around because the one stopped standing behind the chair. Okay, he must be wondering why I cannot get a juice reward because it did wrong. Okay, so we found out a very interesting phenomenon because initially we saw this transgenic monkey could be very stubborn. They don't want to switch to the new choice. However, we do observe the switch from those transgenic monkeys. But the only deficit means those transgenic monkeys more easily return back to the old color. That's even, remember, this task is 100% correct, switch. So once the monkey realized the rule has been changed, he just stick to the new color and for the juice reward. However, the transgenic monkey, they cannot help to return to the old color. So they make more regressive errors here. And this paradigm has a very unique convenience because we can implement this reversal learning paradigm in both monkeys and the humans. So for the monkeys, we just show this deterministic reversal learning paradigm. And when now we implement a human version, of course, the human version is more difficult. We call the probabilistic reversal learning paradigm. So the human version is only 75%. The reward will be given to the subject, and there's 25%. It's just even the subject pick up make the right choice, they cannot get given the reward. Okay, so of course and the beauty is that we can build a unified computation model. It's based on the reinforcement learning, and we build this unified computation model to simulate the monkey and the human behaviors. During limited time, we won't go to the details of this reinforcement model. And during this model, you can see, using this model, we can mimic the monkey behavior and the human behavior both clearly. And we can find a very interesting difference between monkeys and the humans, because it seems the monkeys intend to explore more compared to the new choice compared to the human. So we also did the MRI imaging on this behavior monkeys as well. We trained the monkey how to use the joystick and to make they use the joystick to track the blue square. And once they learn how to use joystick, and we train them doing MRI scan while they're performing this reversal learning task. So you can see, again, this is a two-color reversal learning task, and the monkey can make a choice between these two colors. And right now we're still analyzing the monkey and the human MRI data. This project is still ongoing. So now we switch to the MSCP2 monkeys, and here we use the MRI. We collect the MRI data, especially recent state fMRI data from monkeys. There's 5 transgenic monkey and 11 wild-type monkeys, And we also using human resident state fMRI data from the public data sets, especially related to autism. There's two public accessible data set people called ABID1 and ABID2. In these two data sets, there exist almost 2,000 subjects and 1,000 autism patients and 1,000 healthy controls. And also we use data set from OCD and ADHD as well. Okay. So first of all, of course, we try to compare the pathological circuits of the MCP transgenic monkeys and the autism patients. Okay. And here is the intuition is we suspect or we are expecting the MCP monkey only may, if they share some similarity with the pathological circuits of the human patients, there must be a very small set of human patients. Okay. So that's why we stratify the whole clinical cohorts is consistent of 1,000 subjects. And then we found out only a small set of autistic patients. So it's like 49 out of 500 patients. And then we found that a small set of patients, they share a similar circuit deficit with MCP2 monkeys. And based on this insight, so we wonder if we can build a machine learning model from the feature, we can learn from this transgenic monkey model and then use to the human clinical diagnosis. And then we extract the core regions. There's a feature we learned from the transgenic monkeys and then translate to the human patients. And then based on these 9 core regions, we build up the machine learning classifiers. And then it's interesting because these features learned from transgenic monkeys can significantly improve the clinical diagnosis accuracy. Here you can see the ROC curves. Both the diagnosis accuracy improved from the 60% to the 80%. And also it's very interesting because these features can apply to the OCD patients as well, but not to ADHD. So that we also can analyze the connectivity profiles of these classifiers. And for the ASD, the autism patients suffer more deficit from prefrontal cortex to the temporal cortex. Or for the OCD patients, they suffer deficit more prominently from PFC to the subcortical areas. And also this paper was published last year in American Journal of Psychiatry, and their colleagues will write commentary about our papers. Okay. So there's sidewalk because we're interested in the connective flexibility. So there's a very interesting section we call ventrolateral PFC and this area has a very unique role in the cognitive flexibility. That's why we also using a genetic-based viral tracing to mapping the axon projection from ventrolateral PFC to the rest of the brain. Okay, so let's get the second part of our talk. So the first part that we try to propose the animal model can be used to dissect the clinical heterogeneity and comorbidity. And the second solution is used in the society. It's called big data, of course, integrated with the recent surge of AI. So the UK version, there already exist a lot of papers published based on the UK Biobank data set. And the US version also mostly or initially from the HCP project. So the UK Biobank include almost the half million people's data, including brain imaging and the behavior phenotyping, or even the genomic data as well. So this big data integrates with AI algorithm or AI models as powerful potentials to dissect the heterogeneity as well. And here we want to specifically talk about one specific issue as related to the topic, the central topic of this symposium. And the issue is that most of current diagnoses, especially psychiatric diagnosis, is called a category-based diagnosis. This category-based diagnosis usually diagnoses one subject, one single subject, is in a binary manner. What binary manner? It's either this subject is autism or is not, right? It's yes or no. It's yes or no. But if we're considering the real situation, is most likely, probably, okay. There's one patient or one subject, they suffer, for example, like 30% depression, like 40% anxiety, or 20% social communication, or even 10% sleeping problem, or some other 5% inattention, something like that, right? So it's in contrast to the winner-take-all diagnosis hypothesis, right? It's not a 0 or 1. It's called like a 70%, 50%, or 70%. And so that's called, if people call the quantitative diagnosis are compared to the binary diagnosis, right? So here we like to adopt methods or model using in natural language processing, NLP field. Is, for example, if there is too many documents, or so many documents, and how can we quickly assign labels for those documents? Because different documents have different topics, right? So if we're considering a similar situation, for example, here, okay, there's many documents, and also we can define there are many topics, right? And also this topics is represented by the words, right? So if we consider each document as a single subject, and a topics is like a behavior phenotypes or behavior domains, and then the words is brain connections or brain measurements, and then we can adopt this mature AI algorithm used in topic modeling or using in natural language processing. All right. Okay. So especially in this way, we can quantitatively describe a single document has like a 40% of a sports topic and 60% technology topics. Right. It's quite similar to the scenario in psychiatric diagnosis as well. Okay. So we know a document is a mixture of all topics and each have their spacing weight. Okay, is in a parallel situation, a document is a patient. A patient is a mixture of all the topics. The topics are symptom dimensions or behavior phenotypes, right? And each behavior phenotypes having their own specific weight. Okay, so in this way, we try to build a Bayes, a hierarchical Bayes algorithm or model based on the latent directed allocation, the LDA. Okay, so we're using this whole brain functional connectivity and to construct a factor. This factor is called the latent factors. And those latent factors consist constitutes a single subject. Or in a single subject, you can see from this picture, cartoon plot. A subject 1, they may have a factor 1 is 50%, 15%, the factor 2 is 35%, and the factor 3 is 45%. Factor 4 is 10%, right? So, and this Bayes model can assign each factor with different probabilities for a single subject, okay? So that's we construct, we have this intuition or have this scheme, and then we try to implement in several public data sets. Okay, here again we use this ASD and ADHD cohort data, and then we run this LDA algorithm. I try to identify four factors here, and then we quantify each subject with these 4 latent disease factors. Okay. Of course, this is a data-driven method. For all the data-driven approach, we need to evaluate the data-driven factors, the relation of data-driven factors with the clinical measures or some biological explanations, right? So, of course, the first, we try to correlate this data-driven latent disease factors with clinical symptom scores. That's the first step. And also, we can use the meta-analysis, try to review the underlying cognitive domains of this latent factors. And the third, if we really want to validate this latent disease factors, we want to extend this latent disease factors estimated from ASD and HD to other data set or to other categories. Here we use there's a non-clinical cohorts with anxiety measures, and also we extend to the OCD cohorts as well. Okay, here's the detailed data set we use in this project, and we have ABID2 data set, and also the ADHD data set, and the OCD data set, and also the we call the BSP data set. Okay. And first of all, I will show you the data-driven, data-derived latent disease factors. There are 4 factors, and the 4 factors has a significant difference connectivity profiles, and the different and these factors can be these 4 factors can be variably expressed in individual subjects, and here also plot in the C. And we try to evaluate the associations between these latent disease factors and the clinical symptoms here. And we use the correlation analysis. And here, we found a significant correlation. For example, here, for the factor 1, we found a significant correlation with the behavior regulation index. For the factor 2, we found a significant correlation to the autism, the social communication scores. And for the factor 3, we found a significantly correlated with the anxiety disorder index. And for the factor 4, is significantly related to the interpersonal relationship. And if we plot, we use the connectivity profiles of each latent disease factor and as an input, and try to use a meta-analysis. This input parameters, we use this neuroscience, the website. You can input the connectivity profile of these factors, and then try to find out based on the big data. You can find out the previously published data. Link those factors with specific cognitive domains. And here you can see that for factor 1, is mostly significantly related to the motor movements, premotor, sensory motor. And for example, for the factor 3, it's related to the reward system, it's related to the medial prefrontal cortex, and also related to like social, also emotional regulation. And for the factor 4, it's related to the hippocampus, like conditioning, verbal or working, hypothalamus, and so on. And then the next step, we try to validate this latent disease factors in using an external data set. Here we're using this OCD cohort, and we find out that specifically factor 1 expression in this in individual OCD patients, and interestingly, the loading, the expression level of factor 1 in individual OCD patients is significantly correlated with the white box score of the individual patients. And thanks to the Bayes algorithm, we can evaluate this correspondence in one-by-one patients. And here I give you one-to-one correspondence for all 97 OCD patients. You can see when the factor 1 loading is high and the white box score is high. When the factor 1 loading is low and the white box score is low. So it can demonstrate the good correspondence between the factor 1 loading and the symptom severity of the OCD patients. And similarly, we found factor 3 expressed in a non-clinical cohort called the GSP. In this cohort, it consists of over 1,570 subjects, and each subject may suffer a different level of anxiety, but however, is not clinically diagnosed as anxiety patients. Okay. So it's very interesting. Again, we found out this factor 3 loading significantly predicts the tension and anxiety in individuals. And for this 502 subjects, right, we again observe this correspondence between the factor 3 loadings and the symptom scores. And again, the left two subjects, like subject 132 and subject 283, these two subjects express factor 3 at a large degree, and then they have a very high anxiety score. The other two subjects, they express factor 3 lower at a smaller degree, and then they have lower anxiety scores. And the last step, we try to use this latent disease factor to inform the treatment variability in OCD patients, because among the baseline 97 OCD patients, there are only a small set, like 27 treatment refractory patients, they receiving surgical intervention. So we wonder this factor 1 expression can be used to inform treatment variability in those treated OCD patients. And here, we found a significant correlation between the alterations of factor 1 expression loadings and also the measured white box score change. And here, we also can show you a clear demonstration for those patients who experience a better outcome of surgical intervention. They usually have a large alteration of factor 1 changes. And for the subject, they experience poor response to the surgical intervention. Usually their factor 1 loadings change a little bit. Okay. So in total here, we try to present you a novel quantitative based model to implement a quantitative diagnosis in psychiatric disorder. And here, of course, I have 2 slides to conclude my talk. And again, if we look at the history, we have this very first body image by MRI is collected or acquired in 1971. It's almost over 50 years ago. And if you look at the image at that time, in the middle here, it takes 8 hours to acquire this MR image. They're imaging the body trunk part, and I believe most audience cannot see very clearly what's the internal organs on this image. However, during that time, the one pioneer of the MRI field, Raymond Damian, wrote a fantastic success in on his notebook. And here I want to say is, if you have a more faith, you may enjoy a greater vision, especially for the MRI. For psychiatric neuroimaging is still in a very early stage right now, but we cannot lose faith, because 50 years later, for example, in my lab, just one of maybe thousands of MRI labs around the world, we can achieve 150 microns resolution or 250 microns resolution for the monkey brain, for the structure imaging, for the DTI imaging. So you can, imagine a half century gone, and then the technique, including both hardware and the software development, and then can improve this technique to unprecedented levels. Okay. I'd like to thank all my collaboration partners, and also I want to thank the research funds that support these projects. And again, also in my lab in the Beijing University, we're recruiting PhD students and postdocs and research students at all levels. If you're interested, please send your CV to my email account. Thank you very much.
Okay, thank you very much, Dr. Wang. Okay, so there are many, very inspiring talk and exciting model. So maybe I was starting from a different, so just to get things straight, the 4 factors you got were like model derived. There was no pre-assumptions, right? You got it from the data.
Yes, you're right. Yes, it's completely data driven. Yes.
So the model can obviously predict ASD, ADHD, and OCD because that's the data you input, right?
Yeah.
So basically whatever, so you can in future probably expand your model depending on whatever disease you get, you'll have different factors, and then you can use that to predict the different diseases. Is that how you envisage it?
It's partly true. Okay, it's partly true. Yeah, because of course, that's what I say. A single disease like autism, right, has multiple dimensions. So we can extract latent factors from a single autism data, and these factors can represent many domains, not specifically to autism. For example, maybe this factor just represents a sleeping problem, okay? And then this factor can be expanded to other disease, like sleeping disease. So that's an idea, yeah.
Okay, so there's more questions, but maybe the other speakers have a question, or Dr. Yi. Okay, Yili has a question.
I don't have any question. I really enjoyed the talk, and it's very interesting. It's like in bigger scope than our psychology research, and I think we could communicate a lot later on in our department.
Okay, so
Okay, sure.
Okay, so in the autism in the autism data set, you said only 10% the human patients, they had data corresponding to the monkey data. So is there any genetic data or behavior data that suggests what subgroup of autism patients there are? And another question is, does age affect, how age depend, does your model only work for specific ages of humans, or is development not so important?
Well, yes. When we're in Shanghai, when we're working on this MCP2 monkeys, we try to figure out the ratio of the MCP2 mutated or not only overexpression. Okay, so the MCP2 mutated patients in clinical scenario. So when we collaborate with clinical doctors, we got got some idea. We can screen like 200 clinically diagnosed autism patients, and then only 5 of them may have an MECP2 mutation. So that's about the odd rates in the real world, in the real world. Yes. So yeah, that's MECP2 is just a single gene. It's among of 100, 150 genes in autism disease. So it may only represent a very small fraction of human conditions. Yeah, that's my view.
So how much does developmental age affect the human data?
That's a very good question. But however, we really like to, if we can, we really like to conduct a longitudinal study on those monkeys. But with the time going, we don't have the data, but we have observed those monkeys for over 5 or 6 years. But with the time going, we found out those symptoms is kind of diminished. It's going weaker. Yeah, even for those circular routing inside the cage, they, when they're getting old and this circular routing is kind of diminishing. Yeah, we don't know why, but yeah, that's what we observed.
Okay, thank you very much, Dr. Wang. So I guess that's sort of we're also up to time. Oh wow. Professor Webb is still here. This is
I am. I actually have a question for Dr. Wang if we have a moment left. I'm sorry for dropping in late.
No, sure. Yeah.
Yeah, given MECP2's role in Rett syndrome, and particularly the sort of a female phenotype, because the mutation is I think lethal in males, knockout male mice and stuff, isn't it? But do you think that your findings might have a sex-related difference?
Yes, it's true, but the issue is we only have 5 transgenic monkeys, so it's so 3 of them female and 2 of them male, and unfortunately, the male monkeys, the MECP2 overexpression has very low, so the overexpression level is low for male monkeys, so we cannot link the cause to the sex difference. Because the female monkey, unfortunately, they overexpress like 7-fold high. But for the male monkey, they only like 1-fold. So their overexpression level are different. So we cannot link to the sex difference here. Yeah.
Thank you.
Okay, thank you very much. So and thank you for staying so late. So thank you very much to all the speakers for a very exciting, inspiring session. And I think that concludes our subforum on autism spectrum disorders, quantitative diagnosis. So I guess we can all talk further in detail about potential collaborations, maybe being inspired by each other's talk. And it's lunchtime, or evening time in the US. So thank you very much and goodbye. Bye-bye.
Thank you. Bye-bye.
Thank you.
Bye-bye. Bye-bye.